Now I would request Dr. Shubhra Mishra, convener of the webinar organizing committee, to deliver his formal welcome address.
Thank you, Dr. Chakraborty. Good morning, everybody. Respected Principal Sir, Chairman of the Webinar Organizing Committee, Dr. Bidyut Samanto, respected President Sir, Professor Anjan Chaki, Patron-in-Chief, Webinar Organizing Committee, respected resource persons, other dignitaries, distinguished participants from different academic and research institutions from different parts of our country and abroad. My dear students, my colleagues, ladies and gentlemen. On behalf of organizing committee, myself Dr. Shubhra Mishra, convener of this organizing committee, extend a very warm and cordial welcome to all of you to this webinar entitled Concepts, Innovations and Methods when Chemistry Meets Biology, organized by Department of Chemistry, Kharagpur College, in collaboration with National Academy of Sciences, India, Kolkata chapter. We feel proud to have such adventure. I, on behalf of the organizing committee, wholeheartedly thank eminent scientist Professor Dr. Hemant Kumar Majumdar, President NASI, Kolkata chapter, NASI Senior Scientific Platinum Jubilee Fellow, for extending his hand by collaborating with us and being with us as a renowned orator. It's a privilege for me to welcome you, sir. I must thank Professor Dr. Amit Kumar Das, Professor Department of Biotechnology, IIT Kharagpur, and Professor Dr. Sushant Rai Chaudhary, Chief Basic Research Saroj Gupta Cancer Center and Research Institute, Kolkata, for managing their time for us from their busy schedule. It's an honor for me to welcome both of you, sir. All of our resource persons are very much busy and they are working within a strict timeline. But being kind enough, they have managed to remain present among us today in this webinar. We are in true sense very much thankful to them. I welcome Mr. Sandeep Sen, DSC member, HS Council. I am really thankful to him for his Sir Anjan Chaki, Patron-in-Chief, our Principal Sir, Dr. Bidyut Samanto, Chairman of this committee. I also welcome all the Honorable GB members present here, Teachers Council Secretary, IQAC Coordinator, faculty members of our college and other colleges. A huge number of delegates from different academics, research institutes, industries, ITs and other engineering sectors, and many from others, from different corners of the world for participating in this webinar. I extend a special welcome to them. It's a matter of great pleasure that as many as 1002 participants have registered in this webinar. For us, the most encouraging fact about the webinar is that more than 200 are from abroad. These participants have registered from different countries, namely China, Philippines, Ukraine, Algeria, Sri Lanka, Nigeria, Lebanon, Morocco, Kuwait, United Arab Emirates, Nepal, Indonesia, Pakistan and Bangladesh. I, on behalf of the organizing committee, extend a very cordial welcome to all of them. This webinar is designed to provide an intimate environment to discuss on cutting-edge researches that are ongoing at the interface of chemistry, biology and medicine. This webinar is aimed at addressing different innovations, approaches and their applications in modern chemical biology. In this critical situation, besides many negativity, we have a great opportunity to have such eminent scientists and mesmerizing speakers with us. As an integral part of this webinar, we arranged a state-level online essay competition for both school and college level in collaboration with NASI Kolkata chapter, where we put a very pertinent question to the students whether our earth is safe or not, and asked them to mention the initiatives to be taken by them in this pandemic situation. I hope it stimulates the youth, and I proudly declare that the students not only from our state but also from all over the country participated in this competition. And our jury member faced a lot of trouble to find out the toppers. The result of this competition will be declared at the valedictory session of this webinar. Last but not the least, we believe that the discussion in this webinar would be beneficial to all the students, faculty members, research scholars of India as well as the overseas participated here. With these few words, I conclude here. Please stay with us for next few hours and enjoy the session. Thank you. Thank you very much.
Thanks to the convener for his welcome address. Now I'd like to request Dr. Bidyut Samanto, respected Principal, Kharagpur College and Chairman of the Webinar Organizing Committee, to kindly deliver his inaugural speech.
Thank you, Dr. Indranil Chakraborty. Can you hear me?
Yes, sir. Yes, sir.
Yes.
You are audible.
Okay. Good morning and welcome everyone to this auspicious occasion of National Webinar on Concepts, Innovations and Methods when Chemistry Meets Biology, organized by the Chemistry Department of our Kharagpur College in collaboration with the National Academy of Sciences, India, NASI, Kolkata chapter. It's my pleasure to say that today we have with us Professor Anjan Chaki, President of our college governing body, honorable speakers of this webinar, Professor Dr. Hemant Kumar Majumdar, FNA, FASC, FNASC, FASCT, FTWAS, President of NASI, Kolkata chapter, Senior Scientist, Platinum Jubilee Fellow, Sir J.C. Bose National Fellow, DST CSIR, Indian Institute of Chemical Biology, Jadavpur, Kolkata, and Professor Dr. Amit Kumar Das, Department of Biotechnology and Chairman of Central Research Facility and Head of School of Bioscience, IIT Kharagpur, and Professor Dr. Sushanto Raichaudhuri, FNA, FASC, FASCT, Chief of Basic Research and Molecular Biology, Saroj Gupta Center, Cancer Center and Research Institute, Kolkata. He is also J.C. Bose National Fellow. Sir Sri Sandip Sen, DSC member, HS Council, Dr. Tapan Kumar Pal, governing body member and ex-professor of our college and Vidyasagar University, Dr. Vikas Ghosh, Teachers Council Secretary, Dr. Jyotirmoy Pramanik, our IQAC coordinator, Dr. Shubhra Mishra, Associate Professor of our Chemistry Department and convener of this webinar, Dr. Indranil Chakraborty, Assistant Professor of our Chemistry Department and Secretary of this webinar, Sri Prasanna Dule, Head of our Chemistry Department, Dr. Gagan Chandra Mondal, Senior Associate Professor of our Chemistry Department and other faculty members of our Chemistry Department and other departments, dear students, participants from other colleges, universities and schools, I welcome you all. As you know, all of us are passing through a very tough time due to COVID-19 pandemic and the subsequent lockdown. In the beginning phase of the lockdown, everybody was scared and searching for sanitizer, hand wash, mask and other essential items. In that situation, we thought of preparing and distributing sanitizer among the local people, taking road permission from the subdivision officer of Kharagpur, our chemistry teachers, Prasanna Dule and Dr. Indranil Chakraborty, along with few students of chemistry and physics, came forward and assembled in our chemistry laboratory to prepare sanitizer and handed over to the SDO for distributing among the local people. Due to long lockdown, economic growth, development activities, normal health management, transport, education, everything are disrupted. Our students are also affected adversely. They are forced to stay away from their most sacred place, their classrooms and laboratories. They are unable to meet their teachers and their friends. They were scared about their curriculum. At this juncture, we thought of a routine of online class. Thanks to the advancement of technology and our teachers, our IQAC coordinator, secretary of our Teachers Council, and few more like Dr. Indranil Chakraborty and Dr. Shubhra Mishra of Chemistry Department took the initiative for online class. All the teachers of our college, including the teachers of Chemistry Department, tried tried to help our beloved students by uploading study materials in our college website and different WhatsApp groups. Soon they changed their mindset and came forward with their upgradation in information and communication technology ICT and started online class for the students of all the departments of our college. Although online class is not a replacement of classroom teaching, most of the students of our college, particularly students of science stream who are smart enough with smartphones, upgraded themselves in ICT and joined online class with keen interest, overcoming the barrier of availability of internet facility. However, they are confined to their homes for an indefinite period and are unable to meet their friends and teachers and are worried about their curriculum and exams. Apprehending their mental depression, we arranged online mental support programs for all the students in as many as 9 batches. One clinical psychologist of Midnapur Medical College and Hospital advised in all the programs. Thanks to the IQAC coordinator and the Teachers Council Secretary for full support and cooperation of the Teachers Council and the IQAC. Dr. Indranil Chakraborty also played a major role. Online students mentoring and parent-teachers meeting are also going on. Actually, the lockdown situation has created an opportunity for us to connect distant and distinguished people throughout the globe. We took full advantage of online class and the now popular event webinars to hear out those respected and eminent teachers, scientists, technologists, doctors, social workers, and even monks until now who are out of our reach. Our geography department, chemistry, physics, political science, English, Sanskrit, and history departments, our morning shift,
Okay.
our geography, chemistry, physics, political science, English, Sanskrit, and history departments, our morning shift, NSS unit, and IQAC have already organized successful national and international webinars. More webinars are coming up, but today's national webinar organized by the chemistry department of Kharagpur College in collaboration with National Academy of Sciences, India, NASI, Kolkata chapter is a special one because state level essay competition on how far are you save my earth for school level students and on we have one earth for college and university level students were preceded as a part of this national webinar with a motto to attract the students towards science because only scientific awareness can protect the life on earth. Today's webinar on concepts, innovations and methods when chemistry meets biology is also in the same tune. I would express my heartfelt gratitude to our honorable speakers, eminent scientists and distinguished Professor Dr. Hemant Kumar Majumdar, Professor Sushant Roy Chaudhary, Professor Dr. Amit Kumar Das for addressing on such a pertinent topic which has much relevance in the perspective of spread of COVID-19 virus. Despite their busy schedule, they will be present virtually among us, giving a huge number of participants an opportunity to be enriched, enlightened, and updated with their popular talks. I am also thankful to Sri Sandeep Sen, who played an important role in this webinar and the essay competition. Actually, Dr. Subhra Mishra and Dr. Indranil Chakraborty of our chemistry department were knocking at NASI Kolkata for quite some time to organize a seminar before lockdown. However, all the formalities could not be completed before lockdown, so they are organizing the seminar now in the form of webinar. I hope the webinar will be successful with a huge participation, as many as 258 students from different parts of our country have participated in the essay competition, and more than 1,000 participants from all over the globe have registered for attending this webinar. I express my gratitude to Professor Anton Chaki, President of our governing body, for his continuous support and encouragement and guidance. Thank you, sir. I am also thankful to Dr. Tapan Kumar Pal, member of our college governing body, as well as the headmasters and headmistresses and principals of schools and colleges who took initiative to make the essay competition a grand success. I am also thankful to all the judges of the essay competition who has done a tremendous job of finding the best from as many as 258 write-ups. I must congratulate all the teachers of chemistry department for organizing such a wonderful national webinar and state level essay competition in collaboration with NASI very nicely. We are indebted to NASI also. I hope all the students and teachers who have participated in this webinar will enjoy it. I also hope that our students will be able to come back to their classrooms soon and will be able to organize many seminars in our college with the help of NASI and other organizations. I once more welcome all the honorable speakers and participants. Be safe and be connected. Thank you all.
Thank you, sir, for your kind words. Now, I'd request Professor Anton Chaki, President Governing Body of Kharagpur College and Patron-in-Chief of the Webinar Organizing Committee, to say a few words. Please, sir.
Good morning, Dr. Indranil Chakraborty. Good morning to all of you, and most welcome to our probably 8th webinar organized by the Department of Chemistry, Kharagpur College, in collaboration with the National Academy of Science, India, that is NASI, Calcutta chapter. NASI is the oldest science academy in India, established in 1930 under the leadership of the renowned scientist Dr. Meghnad Shah, who was the first president of the All India chapter. Respected chairman of the organizing committee and principal of our college, Dr. Vidut Samanto, our valuable guest and resource persons, Professor Hemanta Kumar Majumdar, President NASI, Calcutta chapter, and Senior Scientist, CSIR, IICB, Jadavpur, Professor Amit Kumar Das, Department of Biotechnology and Head of the School of Bioscience, IIT Kharagpur, and Professor Sushant Rai Chaudhuri, Chief Basic Research and Molecular Biology of Saroj Gupta Cancer Center and Research Institute, Thakurpukur, Calcutta, which Dr. Saroj Gupta, being a radiotherapist, along with a group of doctors and philanthropists, started in the early 70s decade, I can remember, for the cancer-affected people of Eastern India, with special emphasis on the underprivileged class. At that time, only the Chittaranjan Cancer Hospital has started. I have visited in the 70s, middle of 70s, several times of this history. However, let's make go to Dr. Welcome to Dr. Jyotirmay Pramanik, our IQAC convener, Dr. Vikas Ghosh, TCS, Teacher Council Secretary, Dr. Subhro Mishro, and Dr. Indranil Chakraborty, convener and secretary of the webinar, respectively, Dr. Farid Sheikh, Joint Secretary, Professor Prasanna Dule, Head of the Department of Chemistry, and the senior most teacher of the department, Professor Gagan Chandra Mandal, the participants, dignitaries present, and my friends and the beloved students. It is a proud privilege for me to remain present among the luminaries in the field of the chemistry, or rather I can say the biochemistry today, particularly in the present devastating corona pandemic situation obtaining across the globe and the indefinite lockdown period, which affects every corner of the individual life and the society as a whole. One of the most affected sectors of the country is the education sector in this period, which is thrust to incalculable anxiety and uncertainty of the younger generation. To bring out possible support for the students, Kharagpur College Authority, under the leadership of the principal, combined with some other faculties, come forward to launch a series of webinars over and above the regular online classes to bring out possible support for the students in their academic requirement to combat the emergent challenge. In this context, I like to name some of our young and most enthusiastic faculty members like Dr. Jyotirmoy Pramani, Dr. Subra Mishra, Dr. Indranil Chakraborty, Dr. Shukla Mandal Saha, Professor Subroto Paria of our NSS unit, and some others also, whose zeal and responsibility to our society inspired us to undertake such academic ventures successfully. I put on record my sincere appreciation from heart of my heart for this type of academic venture for the betterment of the students. Now, today's webinar, that is Concepts, Innovation and Methods, under the where chemistry meets by, we can say, which is however, I think we can say this is as a biochemistry or chemical biology is commonly associated with the research in the interface of chemistry and biology. As a truly disciplinary science, chemical biology combines scientific concepts and experimental approaches from both of its parent fields to understand molecular mechanism in complex biological systems, which can be applied in both the clinical and the plant biology also. However, I am confident that the participants will be immensely benefited with the valuable deliveries of our experienced academicians present here with their rich knowledge. I wish all the success of the seminar. Thank you. Thank you all.
Thanks to Professor Anjan Chaki for his kind words. With this, we are at the end of the inaugural session, and with the permission from the chairman, I'd like to switch on to the technical session, which was scheduled to be held on from 10:30 a.m., but it's 10:22. If you permit, we can start the technical session just now. Sir, may I start the technical session?
Sure, sure.
Okay.
All the honorable speakers have already joined, so we can start.
With the permission from all the speakers, we are starting the technical session. In the technical session, our first speaker is Professor Hemanta Kumar Majumdar. Before I request Professor Hemanta Kumar Majumdar to deliver his talk, I would like to give a brief introduction of Professor Majumdar to the audience. Himadri, would you please present the slide where the picture of Professor Majumdar is there? Would you please present?
It is already screened.
Okay.
It is already on the screen.
Okay. Professor Hemanta Kumar Majumdar is President of National Academy of Science India, Kolkata chapter. He is also NASI Senior Scientist Platinum Jubilee Fellow, Sir J.C. Bose National Fellow by DST, ex-Rajaramanna Fellow, DAE, former Head Infectious Diseases and Immunology Division, CSIR, Indian Institute of Chemical Biology, Kolkata. He is an Adjunct Professor, Department of Biophysics, Molecular Biology and Bioinformatics, University of Calcutta. And he is also a former Working Chairman of West Bengal State Council of Science and Technology. Professor Majumdar did his MSc in Biochemistry from University of Calcutta in the year 1970 and subsequently did his PhD in Biochemistry from Bose Institute, Calcutta University in 1975. After completion of his doctoral work, he moved to Albert Einstein College of Medicine, New York for carrying his postdoctoral research work and stayed there up to 1979. Thereafter, he moved to University of Zurich, Switzerland as a postdoctoral researcher and stayed there for 2 more years. After coming back to India in 1982, he joined as a Scientist C in Indian Institute of Chemical Biology and stayed there up to 2012 and retired from his service as Scientist G. After that, he joined Indian Institute of Chemical Biology. Rather, he continued to stay at Indian Institute of Chemical Biology as Raja Ramana Fellow of DAE up to 2016. And after 2016 till date, he is in Indian Institute of Chemical Biology as the prestigious NASI Senior Scientist Platinum Jubilee Fellow. During his golden career, he has been members of several professional bodies and technical committees. I am just mentioning a few of them. He is a fellow of World Academy of Sciences. He is the fellow of all the three science academies: Indian National Science Academy, Indian Academy of Sciences, National Academy of Sciences, India. He is a life member of Indian Science Congress Association. He is a life member and ex-vice president of Society of Biological Chemists. He is a member of selection committee for scholarship of KVPY by DST. He is a member of board of studies of several universities and research institutes throughout India. He is an ex-chairman, expert committee for State Innovation Award, Government of West Bengal. He is a chairman of local chapter of National Academy of Science India from 2019 till date. He is also Chancellor's nominee for the Court of Kalyan University from 2017 till date, and many more committees and professional bodies I am omitting because I shall be short of time if I read all these things. During his career, he has received several awards. I am mentioning some of these. He has received C.V. Raman Birth Centenary Award by Indian Science Congress Association in 2019. Sir J.C. Bose National Fellowship by Department of Science and Technology, Government of India from 2010 to 2016. He has also received Sir Ronald Ross Memorial 57th Foundation Day Oration Lecture Award, Nitya Anand Endowment Lecture Award, Desh Ratna Dr. Rajendra Prasad Memorial Lecture Award, Professor B.K. Bachwat Memorial Lecture Award, Professor Gyanchandra Ghosh Memorial Award, Dr. P.C. Sengupta Memorial Oration Gold Medal by Calcutta School of Tropical Medicine for outstanding contribution in Kala Azar research in 2004. Dr. Hemanta Kumar Majumdar is a distinguished scientist in the country. He was a visiting scientist at University of Texas Medical Branch at Galveston, USA, University of Cambridge, UK, University of Rome, University of Aarhus, Denmark, Robert Koch Institute of Berlin, Germany, LMU at Munich, Germany, and Leiden University Medical Center, Leiden, Netherlands. He has made outstanding contribution towards development of diagnostic and therapeutic agents, therapeutics against the disease Kala Azar. He has been guiding research in the area of parasitology for more than 3 decades and has produced 25 PhDs and supervised 30 postgraduate students. For more than 3 decades, Dr. Majumdar has been working on the molecular biology of Leishmania donovani, the causative agent of visceral leishmaniasis. He has made significant contribution in biochemistry of DNA topoisomerases of Leishmania with reference to development of therapeutics. His area of research includes basic DNA enzymology of topoisomerase, development of anti-leishmanial compounds targeted to DNA topoisomerase, cell death of the parasites induced by DNA topoisomerase inhibitors, and development of kinetoplast DNA-based PCR diagnostics. He has published more than 107 research papers in several reputed international journals and 5 scientific reviews. He has edited 4 books and contributed 4 chapters in books. With this short introduction, I have now request Professor Hemanta Kumar Majumdar to kindly deliver his lecture. Sir, please unmute your microphone, sir.
Okay, can you hear me?
Okay, sir, we can hear you.
Acha. Thank you very much. I'm really proud and privileged that I have been invited by Kharagpur College to participate and to deliver a lecture in this webinar lecture series. I'm thankful to Professor, the principal of that college, for kindly inviting me. I'm thankful to Professor Shubhra Mishra, Dr. Indrani Chakraborty, and other distinguished professors. I feel privileged that I have been invited in this occasion, and I'm thankful to the participants whom I cannot see here because this is a webinar series. As you know, my first slide, please. My first slide.
Himadri, first slide though.
Yeah. See, my first slide, please. The title of my talk is Interdisciplinary Science. I've been requested by Professor Shubhra Mishra that since this is the seminar in the chemistry department, I should say something about chemistry and biology. But I'm not only be restricting to chemistry and biology, I will show you how other disciplines are interrelated. And the students will be benefited. I'm not going to go into deep details of the intricate mechanism of the research work I've been doing, but I will give you some essence of the research, what I've been doing in my laboratory. The next slide, please. My dear students, I think you know the slide, you know the picture of this person. He is Aristotle, and everybody knows that he is a great philosopher, father of philosophy. But you may also know, you should know that he is also the father of biology. He has a friend, his name is Theocritus. Both Aristotle, the teacher of Alexander the Great, and Theocritus, they initiated the biological research in the whole world. Next. In this picture slide, you see there are 5 persons. Now you will say that how this slide is related to my talk. This slide is related to my talk because there are 5 stalwarts in 5 different disciplines, but one thing is common in them. What is the common thing? The common thing is they are human. They are Homo sapiens. And they are like Amartya Sen, economics, Einstein, physics, Roger Federer, tennis player, Jim Watson, biologist, Madame Curie, physicist. They are all, one thing is common, that they are multicellular organism. We are all considered ourselves as organism. And we are multicellular, we are not unicellular. Today, the havoc is going on by one unicellular organism, that is COVID. And is a virus. Virus is an unicellular organism. Bacteria is an unicellular organism. They are called prokaryote. And there are small unicellular organisms like yeast, like protozoa. They are unicellular, but they are not called prokaryote. They are eukaryote because they are unique nucleus, well-defined nucleus. We are also having well-defined nucleus in our cells, but we are multicellular organism. Next slide. Yeah, this is the elaboration of the previous one. Parts of the eukaryotic cell. What you can see here is a schematic diagram of an eukaryotic cells, like my cell, my cell, your cell, and others. This is the schematic diagram. What you can see here is that in the slide you can see that there is a well-defined nuclei, magenta color, and there are mitochondria. Bacteria doesn't have mitochondrion, but here you can see the mitochondria, which are the energy generating organism. There are also other subcellular organelles like endoplasmic reticulum, small endoplasmic reticulum, and then rough endoplasmic, smooth endoplasmic reticulum, and rough endoplasmic reticulum. There's small dots which are called ribosomes, and they carry out the protein synthesis within our cells. So there are so many things inside the nucleus, and we are all having that cells inside our body. Now next slide. Yeah. Now what you can see is another schematic diagram. You can see that is a cell having a nucleus, having subcellular organelles, and they're all important: cell membrane, endoplasmic reticulum, nucleus, vacuole, ribosome, lysosome, mitochondria, Golgi body. There are, see, this cell, we are all having this cell, and the subject, what I'm going to tell you, is the interdisciplinary science. Like a cell can be studied from interdisciplinary perspective, like physicists will see the cell in one angle, chemists will see the cell in another angle, because the chemists are more important in carrying out the reactions. So inside our body, the reactions are going on inside our, just like glycolytic pathway. The glucose is being utilized and generating ATP. There's a chemical reaction. Physicists will see the dynamics of this reaction, and then we are the biologists, we can look at the biology of the cell. Mathematician will carry out the reaction mechanism, and they will see how the mathematics is being used up for carrying out the reaction. Enzymatic reactions are being governed, and we can detect the half-life of the enzymes, the reaction mechanisms of the enzyme, and mathematician's role is that. And engineering and technology, these two things have come up recently, like in the last 30-40 years. When I was in Zurich, University of Zurich in Switzerland, I was given the task of carrying out the genetic engineering. That means I did the cloning of some of the proteins of African trypanosome, which carries out the disease, sleeping sickness. I think many of you have heard of the disease sleeping sickness, which is not a disease in our country, but is a rampant disease in Africa and South American countries. It's a disease for human and also a disease for the cattle. And my job was to develop a vaccine against these particular organisms, which are being carried out by the sandfly. Here in our country, there are many diseases. One of the most important diseases is malaria, which is carried out by the vector mosquito. Mosquito will carry the germ for this mosquito plus the plasmodium, and they bite out the individual and introduce that plasmodium in our body, and then plasmodium will be multiplied in our body. But mind that everybody has seen that if you have malaria, you can see that today you have a fever, tomorrow the fever is gone. Next, after 1 or 2 days, again the fever came. Then again fever gone. Again the fever has come up. So there is a periodicity of the fever. So that is the same thing is going on in African trypanosome. The periodicity of fever and is going on in both these diseases, African trypanosome and malaria in our country. So why that is going on? Because the reason is that see, when a person is infected with malarial parasite, our body will make the antibody against that malaria. Okay, and then they will kill the malaria organisms. But however, malaria changes its behavior. Whenever they change the behavior, the present antibody which is already there cannot recognize the newly developed malarial parasite. So again, we have to generate the new antibody in our body. So it's going on, is a battle going on between ourselves, human body, and the parasite. Next slide. Next slide, please. Yeah, this is a picture of a person whose name is Anton Van Leuven. He was born in 1632 and died in 1723. Why I have shown this person? Because he has developed the handmade microscope, applying the knowledge of physics. He was a man, he was working in a textile industry, and he has interest in the prism and also the lens. So for looking at the structure of the textiles, he used to use the lens, so that he can see the knitting between in the textile. So using that knowledge, he developed this microscope. So physics is being applied for developing a microscope which is being used to see the organisms. And he looked at he has a pond near his house. So what he could do, he used to take water from that pond and looked at the water with that microscope, and he discovered the microorganisms in water, which are called protists. And they are so small you cannot see in the naked eye, but that can be seen with the microscope. So therefore he is called, Anton Van Leuven is called the father of microbiology. So next please. This person is called Mendel. I think the students in the school, the 9, 10, 11, 12, they know about the Mendel's law of heredity. Inheritance is inheritance of biological features that follows the law proposed by Gregor Mendel. And I think everybody has read about the Mendelian law of inheritance. Father has got certain properties which will be inherited to the son. Mother has a property which will be inherited to the next progeny. So that he has developed, he has mentioned, predicted the principles of this heredity using the genetical approach with the corn seeds. So next, please. Now let us come to discovery of DNA. I think you know what is DNA. Everybody knows today. When we were students, we never knew of DNA because I never had any biology background. I had only chemistry, but I had to change into biology in my later career. DNA is a hereditary material. Everybody knows. And not only DNA is a hereditary material, there are other chemicals which are called RNAs. RNAs are also hereditary material. RNAs are hereditary material of small molecules, small organisms like virus. COVID has RNA genetic material. RNA is a single-stranded, DNA is double-stranded. There are DNA which are single-stranded, which are present in viruses also. RNA can be double-stranded, which are present in small viruses. So Friedrich Nietzsche, a German biochemist, found the nucleic acids, which is long-chain polymer nucleotides. And you know nucleotides are base, sugar, and phosphate. made up of sugar, phosphate, and nitrogen bases. Two types of nucleic acids are there, RNA and DNA. Oswald Avery, an American scientist, proved that the DNA is hereditary material. I think the students who are in the class 11, 12, or 9, 10, they have read that experiment on Avery, MacLeod, and McCarthy, why the DNA is called the genetic material. So they have done the experiments on with pneumococcal pneumonia injected into animal, and then have shown that it is the DNA only that causes the death, that causes the death of that animal. So DNA contains 4 bases: A, T, G, C, adenine, thymine, guanine, and cytosine. RNA is having the same thing, excepting instead of thymine, it contains uracil, but there is a difference in RNA has the ribose and DNA has the deoxyribose. Next. Now, this is very important picture. Everybody knows the DNA double stranded. What do you mean by strand? Suppose if you look at the ladder, you can see that there is a bamboo string and there is another one, and there are small steps in that bamboo. But here also the same thing. This is one strand and the other strand, 2 bamboos, and these are the steps. Now you can see here these 2 pictures are the same. Only thing, there's a rotation. Look at this picture. In this picture, you can see one strand is winding the other strand in the right-handed direction. See, this is winding the other strand in the right-handed direction. But here you can see, if you look at very critically, you will see that one strand is rotating the other strand in the left-handed direction. Sorry. So what you can see here, these 2 are the same, these 2 are the mirror image. So DNA is double-stranded with several bases: adenine, cytosine, guanine, and thymine. Adenine is making hydrogen bonding with thymine, cytosine is making hydrogen bonding with guanine, and they are and these are the strands where the phosphate bonds are on the outside. That's why if the phosphate bonds in the outside, that's why the DNA is a negatively charged molecule. Next, these are the further figure of the discovery of DNA. It's a multidisciplinary effort. The discovery of DNA is not done by a single person. Look at the picture on the left. James Watson, he's a biologist. And then Francis Crick. Francis Crick is a physicist. And then Maurice Wilkins, he's a biophysicist, he's a crystallographer. Then Rosalind Franklin is a chemist. She's also a crystallographer. Linus Pauling. And Linus Pauling is a chemist. And he has got Nobel Prize twice. And then Arvind Shargap, he is also a biochemist. Arvind Shargap told that it is the T which will make hydrogen bonding with A, it is the G which will make hydrogen bonding with the C in order to make the DNA strand. And this is the picture of Watson and Crick, a double helical structure. They are discussing this is a very landmark picture in the history of biology. So biology is nothing but is always the discovery in biology comes with the help of physicists, biophysicists, chemists, and biochemists. Next. This is the person whose name is Alexander Todd. We read the book of Alexander Todd, Western Todd, in our MSc biochemistry classes. He got the Nobel Prize in Chemistry in 1957. Isn't it interesting to have a Nobel Prize in Chemistry for the work in biology? Polynucleotide phosphorylase, DNA, this nucleotides and nucleosides discovery has been done by Lord Alexander Todd. I am very proud and privileged that I went to Oxford and I delivered a lecture where Lord Alexander Todd was present, and we had a dinner in that after that lecture. So I'm very happy, and Alexander Todd has so many Indian students in our country. So next, see, this is a picture of a DNA. So I think, suppose you think about a rubber band, okay, garter. So if you make, this is the structure of a rubber garter, okay, on the top. Now if you can make the rubber garter, if you wind the rubber garter, you can make the structure like this. You can do that. If you do it more, you will have this type of structure, more and more entangled. Finally, a rubber garter which is circular, it can be made a linear by just twisting the garters. They are the same thing. Only what is the difference? Difference is in their morphology, their structure. They can be made. Their physical structure is different. But DNA is a sequence which encodes the RNA and the protein. These and these two are the same, only the physical structure. Now, if you take a rubber band and you just throw it from top, and the floor, it will fall in the floor. Now, if you take this rubber garter and make a structure like that, and tangle it and then tangle it, and you can see the structure will be like that. And if you fall it from the top, it will also fall in the bottom. But the time taken by this one to fall and the time taken by this one to fall will be different. This will fall faster and this will fall later, slower. So that is because of the Bowen density of the same thing. So now if you can take this type of DNA structures and run a gel electrophoresis, I think many of you have, not the school students, but when you will come to undergraduate college and the postgraduate college, you will see the gel polyacrylamide gel electrophoresis or agarose gel electrophoresis. You can do the electrophoresis of these DNAs, and if you, after doing the gel electrophoresis, and then if you stain with a fluorescing dye, like ultraviolet fluorescing dye, like ethylene bromide, the DNAs which were migrating in the gel differently because of their buoyant density, because of their structure, they will migrate in the gel differently and they will be fluoresce differently. So the same, this DNA and inside those DNA are the same in terms of the chemical composition, but in terms of the morphology and their structure, they are different. So their migration is different. This is very important in the biology. And this type of structure has been done, experiment has been done by the chemist. So it's a morphological. So this is called highly supercoiled, and this is highly supercoiled, means this one, the bottom one is called the highly supercoiled, and the top one is called no supercoiled, it's only relaxed. They are the same, they contain the same composition, same sequences, but inside the cell, if you can look at a human body, if you look at a cell inside in our body, in one cell, the amount of DNA which is present inside the cell, and you know that between the two bases of a DNA, there is a difference of 3.4 angstrom. What is angstrom? 1 angstrom is 10 to the power minus 8 centimeter. Now we have, you know that every person, we have, how many cells are there in our body? Trillion cell, 1 trillion cell. And 1 trillion cell, one cell contains 23 pair of chromosomes, everybody knows, 23 pair of chromosomes. And every chromosome contains the DNA. And if you calculate the total amount of DNA inside our nucleus, it will be enormous amount. It cannot stay inside the nucleus in a cell. So they have to take the configuration like that so that they can be allowed. They can stay in the nucleus of that cell. That is the way they stay inside the cell. Now the question, why I'm showing this one? Because these configurations are called same DNA, different configuration. They are called topological isomers. When a cell is multiplying, the DNA of the cell will be multiplied. When the DNA of the cell will be multiplied, you know that the DNA is a double-stranded DNA. They have to be unwound, and if you do not unwind the DNA, the rest of the reactions like transcription into RNA cannot be taken place. All enzymatic reactions are there. They will be carried out by a multi-enzymatic system. Next. See, this is a cartoon picture. Just what you can see, this is a DNA. It's a double-stranded DNA. You can see one strand is winding the other in a right-handed helix, and now you can see here these two strands have been separated, and this is being replicated. Okay, one DNA will be replicated into two. How? So two strands will be four strands altogether. So one DNA will be replicated into two from one side, and that is called the origin of replication, and the fork is moving in that direction. Okay, inside our body, inside our cell. So one DNA is replicated into two DNA, and that is called DNA replication. So you can see that one single DNA has been replicated into two in our body, inside our cell. Now the question is, DNA in our body. See, I'm talking and talking, so I need the energy. If I need the energy, I need the synthesis of the ATP. If I need the synthesis of ATP, adenosine triphosphate is an energy generating molecule, then transcription is required, and then the protein has to be synthesized. Now what you see, then the DNA is transcribed into RNA. So you can see the DNA. If you want to do the transcription, DNA has to be unwound and then become single-stranded, and one single strand will become the template, and the RNA is the red structure is the RNA, so RNA will be synthesized. And this is the green one is the cell wall. Okay, so what you can see that the RNA this is not the cell, is the nucleus, is the nucleus wall. So RNA will be synthesized inside the nucleus of the cell, and then RNA will be transported into the cytoplasm. This is the outside area, is the cytoplasm. This is the RNA red one, and the RNA is coming out from the nucleus to the cytoplasm. Replication is going on inside the nucleus, transcription is going on inside the nucleus, and then transcription, RNA is being transported into cytoplasm, and that will be translated with the help of ribosome in the cytoplasm. So translation of protein synthesis is being taking place. Whatever energy I am spending here, this is for. But human DNA cannot be replicated once they're complete. They're grown up. So it's undifferentiated. But inside the cell, whenever you take up some liver from our body, and that liver will be regenerated. So replication is good, but unicellular organisms, they can be replicated as and when you are allowing them to replicate. You give them food, they will replicate. So transcription of DNA to RNA to protein, this is the central dogma and the backbone of molecular biology, helped by physics, chemistry, and other mathematics, and also other areas of science. Next. See, this is a very important discovery of insulin. Insulin is discovered sometimes in necessity of this protein. Insulin is a hormone. It's a small peptide. This hormone, necessity of this hormone has been indicated by Paul Langerhans. Paul Langerhans, sometimes in 1800, he has shown that in our pancreas there are some cells which are called islets. In the pancreas, some proteins or peptides are being synthesized which are called insulin. That is important for controlling the sugar, and that That was in 1800 something. And these gentlemen, Bunting, Best, Collip, and MacLeod, in Canada, they have all for Professor Bunting is the person. Professor Bunting is the person who discovered this with the help of his student Charles Best. So there's an institution in Canada, Bunting and Best Research Institution. Collip and MacLeod was the head of the division. They discovered insulin sometimes in 1923 or something like that, 22, 23. Next, see, Frederick Sanger determined the sequence of this insulin. As I told you, there is a peptide, not a is a protein, but peptide. It has two chains. One is having 21 amino acids. These are called, the small black beads are called amino acids. Defined. There are 20 amino acids are there. Here in the A chain there is a 21 amino acids, and the B chain there is a 30 amino acids. So 30 and 21 is 51 amino acids. Looks like a necklace comprised with several amino acids. There are 20 amino acids are there. So Frederick Sanger, he determined the sequence chemically of this insulin. So that was in 1953. He did that, and he got the Nobel Prize in 1958. Next, please. See, look at the picture. Try to remember the picture of this gentleman. Fred Sanger was awarded Nobel Prize in 1958 for his work on the structure of the protein. Not only insulin, the same procedure can be followed to understand, to determine the structure of other proteins. So look at this. Try to remember his picture, because I will show his picture again. Next. See, as I told you, that DNA to RNA, RNA to protein, and this is the central dogma, DNA to RNA, messenger RNAs. Here is the messenger RNA, is a dotted one, is a gap, is shown with a red line. Okay, these messenger RNA has to be translated into protein. Who does this translation? Who are the enemy? Who are the armies? Armies are the transfer RNAs. A part of this transfer, there are three types of RNA: ribosomal RNA, transfer RNA, and the messenger RNA. There are other RNAs also. So this is the transfer RNA, which are very small, 71 nucleotides long, and you can see here there is AGU. There are three nucleotides. These are called codon. Okay, and here you can see UAC. That is called also a codon. And this codon UAC and this codon AGU. The transfer RNA will sit on that codon with a complementary nucleotide, because transfer RNA, messenger RNA is also having nucleotide, transfer RNA is also having nucleotide. So AGU has a complementary in the transfer RNA UCA, and messenger RNA UAC has a complementary with the AUG. This is the initiation codon, and the transfer RNA will this is the AUG is the initiation codon, but here the UAC is the codon in the messenger RNA. So they will be read by AUG in the transfer RNA, and transfer AUG causes will be responsible for here is tyrosine. Look at this. A, U, and G is this first base, second base, and the third base. U C U is a phenylalanine. U C is a serine, like that. Okay, so this is the genetic. The complicated genetic code has been developed and discovered by. Next slide. See, these three people, Robert Holly, Herkevin Corona, and Marshall Nirenberg. These three people, 1968, they got the Nobel Prize because of this prediction of the genetic code. There are 4 nucleotides, A, T, G, C. 4 nucleotides. If you do the permutation combination, there will be 64. If you use that codon triplet, there will be 64 codons. Yes, there are 64 codons, but amino acids are 20 or 21. So these 20, 21 amino acids will be taken up by the 64 codons. So different codons can take the same amino acids. Okay. So Robert Holly, Horgovind Khorana, and Marshall Nirenberg, they got the Nobel Prize for predicting this. And chemically, in the laboratory, they have shown that yes, if you have in the mRNA, messenger RNA, U, it will be polylysine. That will be a polyphenylalanine. Phenylalanine will be taken up. Next. See, another landmark is DNA replication. Major experiment was done by the chemistry people and the physics people. DNA replication, look at the double-stranded DNA. One strand is represented yellow by red and the other one is blue. So this is the semi-conservative replication. When one strand is this and this is 2. So semi-conservative replication, the replication bubble is taking on, is like the unwinding of the replication 4, and the 2 directions the replication will take place. I have done work on this DNA replication while I was in University of California at Berkeley. So I have a major work is a polyenzymatic reactions. Excuse me. Next. See, in 1959, 2 persons, Severo Ochoa and Arthur Kornberg, these 2 people are very much involved in the discovery and the prediction and the enzymatic reaction of DNA replication. For the discovery of the mechanism of in the biological synthesis of ribonucleic acids and deoxyribonucleic acids. Ochoa has so many students in our country. They are very well settled in our country, and they are also very influential and very well-known scientists. Arthur Kornberg, he has done a lot of work on DNA replication, how the replication machinery is moving, what are the enzymes involved in replication. 14 enzymes are involved in DNA replication, and that was predicted by Arthur Kornberg. Arthur Kornberg got Nobel Prize in 1959. His son has also got Nobel Prize. His son name is Roger Kornberg. He got the Nobel Prize sometimes later. That is all. You are wrong. He has shown a very important discovery that nucleosome, that means DNA, how DNA is enwrapped inside our cell with the help of the proteins called histones and other non-histone proteins, and that the structure of that protein and involvement of histones has been predicted by Roger Kornberg. Next slide, please. This is the discovery, gene cloning and recombinant DNA. In 1980, I had been to University of Zurich, Switzerland to learn the recombinant DNA technology. Recombinant DNA technology means, it's a very simple example. Suppose you have a diabetes, you need insulin, that has been shown, established, and insulin is not a big protein. And whenever you need insulin, what do you do? Animal slaughtering is being done. Pancreas is being taken out from the animals, like bovines, and then you homogenate the pancreas, take the supernatant of the pancreas, purify it, and inject it into the diabetic person. That was the treatment was done. And that was continued for quite a long time. But if you want to produce insulin, you have to slaughter the animal. Is it really is it good? No. So a technology has come out. Fred Sanger, he has shown the sequence of that insulin, chemical synthesis, chemically he has shown. Now synthesis of insulin has been done chemically, and then this insulin has been introduced into bacteria. So if you introduce into bacteria, in a bacteria is a unicellular organism. The amount they can grow very easily if you give them food. So, and bacteria also contain some extra chromosomal element which are called plasmid. So plasmid is this. Look at that. This is a plasmid, and this is the DNA of the bacteria, just a part of the DNA. So this is the plasmid, and the plasmid is extra chromosomal element. They grow in as and when the bacteria is growing, plasmid is also growing. So what has been done, the technology is you take the chromosomal content, say insulin is present in our human body, take the human DNA, isolate the gene for insulin, and then, or you can synthesize the gene for insulin, and then you put it into bacteria, plasmid. So this is the plasmid DNA, and you put in this insulin DNA, and then it will call a recombinant DNA, plasmid DNA and the insulin DNA, foreign DNA. This is called, and then they are introduced into bacteria, and the bacteria will be multiplying. Once the bacteria is multiplying, this insulin will be synthesized in our bacteria, and then you can get enormous amount of bacteria, enormous amount of bacterial lysate, enormous amount of insulin we can isolate from the bacteria. So that is called recombinant DNA technology. Next. So this is the picture of scanning electron microscopy of a bacteria E. coli. And this is the plating bacterial colonies in agar plate. Next. See, Paul Berg and Walter Gilbert and Fred Sanger. I told you to remember this picture of Fred Sanger. This is Fred Sanger again. And Paul Berg is the person for fundamental studies on biochemistry of nucleic acids with particular regard to recombinant DNA. He is the person who has the pioneer of this recombinant DNA technology. Walter Gilbert is a mathematician, and Fred Sanger is a chemist. So these two and Paul Berg together, they are the person who are involved in this recombinant DNA technology. They all of them got the Nobel Prize. Look at the picture next. See, this is Paul Berg, Nobel Prize in Medicine 1980, and the same Fred Sanger who got the Nobel Prize in 1958. Again, he got the Nobel Prize in 1980, and Walter Gilbert is the mathematician. So biology is totally dependent on physics, on mathematics, and medicine. Arthur Kornberg, I told you, he is a doctor, medicine. Next. See, so I told you an example of only insulin. There are so many recombinant proteins which are very important in our human body. Human growth hormone, recombinantly it is produced. Human insulin, follicle stimulating hormone, factor VIII. Factor is a hemophilia. You know, hemophilia is a disease. Is a disease for the male, not for the female, because whenever there is a blood coming out from your body, it will not be coagulated. The factor is needed. Factor VIII is important, which is called Ekman's factor. That is missing. There's so many, but they can survive if you inject the factor VIII. So it is very important. So factor VIII is also being produced recombinantly. Erythropoietin and hepatitis B vaccine. So these are the proteins which are synthesized using the recombinant DNA technology. And Eli Lilly, Serrano, Bayer, they are the company. They synthesize, they produce this protein. So I will take some more of this. Okay, next. So this is a important discovery. I think I will take only 4 or 5 minutes more. PCS, polymerase chain reaction, an outstanding discovery. Next. See, polymerase chain reaction means chain. If you, that means this is a DNA, template DNA. Okay, this is a template DNA, and now if you want to replicate, it will be replicated once. If it is replicated, it will be replicated into 2. And this will be again replicated into 2. This will be replicated into 2. This will be replicated into 2, and so on. So therefore, is a chain reaction. Polymerase, you know, DNA replication is going on by the enzyme DNA polymerase. Okay, And in our system, biological system, our body temperature is 37. So whatever reaction is going on in our body is 37 degrees centigrade. So DNA polymerase reaction, all reactions were being carried out so far at 37 degrees centigrade. But polymerase chain reaction, what you do whenever you take a DNA, if you want to synthesize, one strand will be replicated here in 2, another strand will be replicated here in 2. You have to separate the strand. If you want to separate the strand, you have to bring the temperature, you have to incubate at a high temperature. So this high temperature is something around 80, 90 degrees centigrade, so that the DNA strands are being separated. And then there is annealing temperature and then the extension temperature. So an enzyme is required, which is isolated from thermophilous bacteria, the bacteria which are grown in the at extremophilic conditions. So there are Taq polymerase, Thermus aquaticus, is a bacteria. From that bacteria, DNA polymerase is being isolated, and that is used in polymerase chain reaction. I remember when I was doing my, conducting research in my laboratory, one professor came from Cambridge. He showed that polymerase chain reaction. Now PCR machines are available everywhere. At that time, he showed the PCR in our laboratory, not with a PCR machine, but with 3 different water baths. One is a normal temperature, and the other one is 95 degrees centigrade, and the other one is 50 degrees centigrade. Taking out the tube from one water bath to another water bath to another water bath. That is the way he was going, and the reaction is going on for 3 hours, 40 cycles. So every time he was doing like that for 3 hours. So that was the way it was done. Now PCR machine has been developed, and you know the COVID reaction by RT-PCR. RT-PCR reverse transcriptase and then polymerase chain reaction. Next. So this is a picture of 2 gentlemen, Carey Mullis and Michael Smith. So this Carey Mullis and Michael Smith, they got the Nobel Prize in 1993, and they did this reaction sometimes before that. See, they were from United States, and we love United States because their attitude is something very, very different, and it's really appreciated. I give you a very interesting story, just 1 minute. Carey Mullis was invited to deliver a talk. Today I have been given time for 40 minutes. I made 45 minutes or something. So Carey Mullis was invited to give a talk, and the time was 20 minutes. So he was giving the talk, and then the chairman was telling to him that, Professor Mullis, you have already completed 10 minutes. You are left with 10 minutes more to tell about your science. Then he was talking and talking at 15 minutes. After 15 minutes, he was told by the chairman, you have completed 15, we have only 5 minutes more. So you have to say whatever you say in 5 minutes. Then he has completed 20 minutes. The chairman said, Professor Mullis, you have to come down. So Professor Mullis has to come down without giving his lecture, because he could not finish his lecture within 20 minutes. And the chairman said, there are so many other speakers are waiting for you, and I don't want to spoil my seminar, so please come down. And he came down. That is the way United States is going on. Can you tell me, sir, you come down after my time is over? You cannot, because this is our India. Anyway, I'm sorry, I tell you, I will be finishing 5 minutes. Next. So this PCR is a wonderful discovery. The discovery of DNA is a real discovery in biology and chemistry. Application of PCR discovery, again, the same important discovery in human civilization is important, because you can determine the total number of DNA nucleotides in human body. So many that you can determine with the PCR and then sequencing. Okay. Application: genome sequencing, diagnostics, genetic disease, infectious disease. I work on infectious disease. PCR is very, very important on that. Next. In 2-3 minutes, I will tell you. I have been working on this Leishmania parasites, which causes kalajar in our tropical and subtropical countries, including India. This is schematic diagram of that parasite. It's a single cellular, is a unicellular organism. This is the nucleus, and this is the flagella, and this is the mitochondria, which is called kinetoplast. That kinetoplast is very, very unique and important. This is the form which is carried by the vector sandfly. Sandfly bites that in our human body, injects that parasite, it goes in our body and is converted into this circular form which is called amastigote. This is the form which is present in our body. Next. See, this is the disease caused by this leishmania. Visceral leishmaniasis, post colors are this dermal leishmaniasis, cutaneous leishmaniasis, mucocutaneous leishmaniasis. A person is infected with visceral leishmaniasis, kalajar, he is treated, he is cured, he went home, 7 or 8 years later he comes back, that he develops nodule in his face. That means, just like leprosy, it stays in our body. Manifestation is not there. The same is like Mycobacterium tuberculosis. They can stay in our body hidden. Leishmania is also like that. Next. So I will show you some of the diagnostics. Next. I told you that the mitochondria has a DNA which is called kinetoplast DNA. It's a huge structure, and that there are in the mitochondria there are 10,000 circular DNA molecules. This is electron microscopic picture, and the small circular DNA molecules they are looking like that. There are 10,000 circles are there. They can be used for this diagnostics. Next. Next. This is the total DNA present in inside the mitochondria of that organism, of that unicellular organism. 10,000 circular DNA, all small circular DNA, huge DNA. They are very important. and you know, you have heard of this RNA editing that has been discovered in this kinetoplast DNA. It's very important. Circular DNA molecules are the same in terms of their sequences. They have two regions, conserved region, variable region, and we have isolated these circles, determined the sequences, and we developed the primers for PCR. And next, this is the kDNA-based diagnostics. So you can see that from different patients, we have isolated the DNA from the blood, and then isolated the DNA, used the DNA as a template, and did that PCR with the help of the information from the kinetoplast DNA, small circular DNA molecules, and you can see that these are the positive. So this kinetoplast DNA-based PCR is very successful for determining the infection. The same thing is being applied in case of COVID-19 infection. Here is the RNA, because the COVID is a RNA virus, single-stranded, and that RNA is being replicated into DNA by reverse transcriptase. That's why RT, and then the PCR is going on. So similar type of thing will be seen in the diagnostic system. So next. Any biotechnological application that uses any technological applications that uses biological systems or living organisms to make or modify products or processes for specific use. So biotechnology, application of technology in the biological system is called biotechnology. Agricultural biotechnology, food production is a biotechnology, medicine is a biotechnology. Next. So I think I have taken a good amount of your time. Sorry, I am not Kari Muli, but anyway, please excuse me for taking too much time, not too much, but sometime. These are my very successful students. They are all very well settled in the country, and they are very. This is me when I was young, and this is me when I'm old. Okay, so it's nothing but morphological transformation during the ages. So thank you very much. Thank you very much.
Thank you, Professor Mojumdar, for your very wonderful lecture and very highly informative lecture. And the participants have typed some questions in the YouTube chat box. And for shortage of time, I am asking one of them. Sushmita Maji, he asked how the chemistry or chemical reactions affect our body or biology.
How the chemical reactions, chemical reactions inside our is not a chemical reaction like sulfuric acid plus zinc equal to hydrogen plus zinc sulfate. It's not that type of chemical reaction. Chemical reaction means, say for example, glucose plus is the catalyst. The catalyst is a enzyme, say phosphorylase. So if you take the glycogen, which is the polymer of glucose inside our body, glycogen plus phosphorylase, the glucose will be coming out, and that glucose will be metabolized, glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to fructose 1,6-diphosphate. All the chemical, these are called biochemical reactions. So it's a chemical reaction, but going on inside our biological system, that's why they are called biochemical reactions. So chemical reactions are there. Yes, we are parallelly maintaining the chemical reaction in our body.
Okay. Sir, one more question. What are antigens and antibodies, and what are their functions?
So I tell you, see, for example, suppose you have been affected by malaria. So what malaria means in an organism, Plasmodium vivax or Plasmodium falciparum carried by the vector mosquito. Mosquito will bite you and they will introduce that organism, Plasmodium parasite. That Plasmodium parasite is enemy to our body. It has to be fought back. This enemy is called an antigen. The proteins of the surface of the surface proteins of the malarial parasites are working as something which will be detected by our body, by our immune system, and our immune system will develop a compound which is called antibody. That antibody will fight with the antigen, means the proteins, surface proteins of the malarial parasites. So foreign substances are called, generally in a simple language, foreign substances are called antigen, and our system will fight that antigen by developing something which can fight that antigen, and that developing something means antibody. That antibody is a protein, and that antigen is also a protein. So antigen-antibody reaction is going on and is being precipitated. Okay. Any protein you inject in our body will fight. Any protein, they will prepare antibody against that protein.
Okay, I think the participants have got their answers, and thank you very much once again, sir, for your effort that you have rendered here, and for your how you extended your helps of cooperation in conducting the seminar, and we are once again thankful to you, sir.
I have a one suggestion. Please, you give, if anybody is interested, you give them my email number. They can ask the questions through email. I will answer.
Okay. Thank you. Okay. I shall send your email number in the YouTube chat box and ask them to send the questions to you.
Okay.
Okay. Thank you, sir. Now, our next speaker is Dr. Amit Kumar Das, Professor, Department of Biochemistry, Indian Institute of Technology, Kharagpur. I think sir is ready, but before I request him to start his speech, I shall take the privilege to introduce Professor Das before the audience. Professor Amit Kumar Das is a Professor Department of Biotechnology, and he is one of the finest structural biologists, and currently the professor in the Department of Biotechnology, Indian Institute of Technology, Kharagpur. He is also the head of School of Bioscience and chairman of the Central Research Facility Life Science Division at IIT Kharagpur. Professor Das completed his MSc in Physical Chemistry from the University of Calcutta, followed by PhD from Shaha Institute of Nuclear Physics, Kolkata. He did his postdoctoral research at the University of Oxford, UK. He was involved in many international collaborations in the field of structural biology and genomics of M. tuberculosis and S. aureus. He visited many international laboratories to set up research collaborations. Professor important proteins from pathogens involved in virulence and pathogenicity. He is also engaged in structure-based drug design to combat tuberculosis. So far, he has submitted 65 protein structures in Protein Data Bank. He has published more than 135 articles in international journals of repute. So far, 25 students have been awarded PhD degree under his guidance, and few more are on roll. With this short introduction, I would now request most humbly Professor Amit Kumar Das to deliver his speech. Please, sir.
Thank you. Thank you very much. Can you hear me?
Oh, yes, I can hear you.
Right. Okay. Right. So thank you very much for giving this opportunity to deliver this talk to the students, where probably I can give some glimpses of how this structural biology can correlate the biological function. Whatever Professor Majumdar told, he almost covered everything, and mostly he covered very important molecules involved in the biochemical reactions. And I will take one of such molecule and try to give some structural overview of those things. So my title of the talk is Deciphering Life at the Molecular Level. That means life means its biology, and molecular levels means we want to see really how these molecules are structurally correlated for their functions. That means structures at atomic resolution. And since the talk is on concepts, innovation, and methods, so I will give some concepts and where innovation can work and how the methods can be developed further for the progress of science for the benefits of the society. So if we look forward, the old sayings said by few Nobel laureates, then we will see that there is no special talents, but you require persistence. And to prove any scientific theory, probably you require failure, failure, and failure until you get the success. So month and years, 99 times, you will get the false result to prove your concepts. That is by the Albert Einstein said in 1921 in Nobel talk. And always most of the thing we know that the opposite of correct statement is always a false statement, but he thinks in a different way that a profound statement can be overcome by another profound statement. That means science is dynamic. What you today think is correct may be wrong in the future, and new theory will crop up. And Richard Feynman, very famous physicist and very famous lecturer also, orator, he told in the near future of the developments in biology will make problems like no one else ever seen before. And probably you are witnessing this thing in the current pandemic situation. And the Peace Nobel Prize given in 1922, and he told that difficult is what takes a little time and impossible is what takes a longer time, little longer time. So now in the rat race of this science, most of the people counting their numbers of the papers and looking for the shortcuts to get some result, but this will not really help to grow science. So we have to take some difficult, challenging and impossible projects to come up with new idea, and where this biology and chemistry, physics, engineering can breed together and give a new subjects, what people call the name biotechnology, nanobiotechnology, biomechanics, bioelectronics, biosensors. So you see all these things, they are now tagging a baseline of bio to understand. If you look physics, chemistry, math, they are mostly saturated, but this biology is dynamic and still we do not know about this process of these biological systems. In 1998, when the human genomes were published, people thought that they have owned the game and probably they can dictate now human life. But still today, if you see, it is not under the control of the human minds. Still, you have to look and handshake with few more subjects to understand this biology, and people are now moving towards this artificial intelligence, machine learning, deep learning to protect the life of the system and to enhance the fastness of this discovery. So whenever we do this work, basically most of the students, they do not think about these parameters, because students read their books but hardly they observe anything. And in current scenario, when people are, basically students are learn to read books and books and hardly they look around to see the nature, probably the observation power of these students are very less, and as a result no new things come up. Every result actually is given in the nature. You have to just find out what is there and how can you explain that and how can you import these observations into your thought process. So parameters, we mostly work on time, and people know this time, minutes, seconds, hours. But if you want to really understand very transient processes of this biological system, then probably you have to go to microsecond, then nanosecond, femtosecond, picosecond, at that level. That means in 10 to the power minus 9, minus 12, minus 15 seconds to record the happening. And at this moment, probably it is very difficult to record all this biological mechanism at this time scale. Because if the reaction happens very fast, you cannot really lock the reaction. And that's why you need to develop some kind of instrument so that you can really see the dynamics of the reactions inside your cells, which is still not possible today. And so it requires kinetics, and mostly these reactions are governed by kinetics or the thermodynamics. We have to look for what? If you look at, so I got one question from the last speaker's presentation, that what is the difference between the chemical reactions and these reactions happening in the biological systems. If you see in biological systems, the reactions are not alone happening, single reactions. They are always coupled with some energy providing reactions. So whenever you join these 2 or 3 reactions, then it is energetically favorable, and we call these are the coupled reactions. And that's why energy is a driving force to govern all these reactions. So time scale, probably I have told that how much you can measure, and probably in chemistry also you have learned about the stock flow techniques to lock some reactions at very fast rate. Now I will come to another dimension, which is distance. Whatever we see in our naked eye, that we can believe, because we believe that seeing is believing. Until you see it, you really cannot believe it. So distance matters. And this distance means if you go gradually, so Professor Majumdar told about microscope. Yes, you can see some microorganism under the microscope as long as it is microorganism. But whenever you are going really to the molecular level, probably microscope will not help that much, because it's a concept of resolution, how you can see these molecules underneath the microscope. So you require visual perceptions that I will talk about in the structural biology. How can you really see these molecules and their structures? And obviously, third part, most of the students, they actually ignore this precision and accuracy. Many people, they do the experiment, get some result and they report it. But whether they are reproducible or how far they are reproducible, that is not properly tested. So accurate means, let's say you have a value of 50 and you are getting the answer is 49. So you are nearly accurate. But if you see in terms of 49.01, you are more precise. So it means that you are measuring the parameter at the second decimal point. If you want to say it's 49.00001, then basically you are measuring at the fifth decimal point. Do you have really such instrument to measure this value in this fifth decimal point? If you don't have, really you are not really going towards the precision. So probably this is the first chapter most of the time in the class 10 or class 12 levels of book, but this is highly ignored by most of the students. So these three parameters are very important in performing any experiment. And then whenever we are talking about these molecules, we have to see few things. Molecules are not just like a straight line. And even a straight line is a geometry that may be a transient part of the molecule, but you require really the three-dimensional geometry of this molecule. So geometry is very important in deciphering the function of these molecules. And whenever you have three-dimensional proper geometry, then probably you will end up with some particular chemices or groups where particular dimensions, width, length, depth, etc. matters, and this will be so specific reactions so that it may be a stereo specific in nature and lock and key model will work. So it is very specific, and then also product will be very specific. It may be stereo selective or regio selective. So those are chemistry students, those who are chemistry students, they will probably understand what is meant by the stereo specificity in the chemical reactions, stereo selectivity and regio selectivity. So to do all these things, probably you need to understand the molecular mechanism. So you can understand the molecular mechanism only when you will see the three-dimensional structure of a protein. And this passes through many transient state. Whether you are able to take the snapshot of this transition state and to calculate this energy at this level, that is a challenge for nowadays discovery. So my talk will encompass mostly on the structure, function and allosterism. So if we see, we require some sample, and probably this sample you have to purify in high level, and you have to produce in high quantity, and you have to measure their quality assessment. Whenever any molecule is sold in the market, then you have to already do the quality assessment. Any drug, whatever you are taking, you have to do the quality assessment. These drugs are mostly the small molecules, though there are many molecules or therapeutic proteins are available, like insulins, whatever Professor Majumdar told. That's very also important. Vitamins also are important. They are small molecules, but many cofactors are generated from these vitamins. Salts and ions, it looks only very small ions, but they are very important, because if you look for the manic depression, then lithium ion is the only therapy. So lithium ion works on the IMPase, inositol monophosphatase, and this position of this lithium ion will dictate how your manic depression can be repaired. Water is very important molecule, which gives really structures of all these molecules, especially these macromolecules. Lipids and fats are important in biological cells because they make the membranes. Lipids are involved in the membranes, and membranes are the protein lipid layer. So you have to produce these lipids in the biological system. And whenever we are talking about macromolecules, this macromolecule mostly encompasses these proteins or enzymes, DNA, RNA, and carbohydrates. Carbohydrate is very tough subject. It has different kinds of conformations, and recognition is done mostly by these carbohydrate molecules. If you look for this blood group, that is according to the glycation on the protein molecule. DNA, RNA, the structure is quite simple. It is double-stranded, so not much various structures you will get. But in proteins or enzyme, you will get different folds and different kinds of structures. So I will concentrate basically on proteins and enzymes in the later part. So this is the era of interdisciplinary research where amalgamation of chemistry and biology is necessary, though other physics and engineering are almost essential part of this amalgamation. So use of scientific concepts borrowed from both chemical and biological sphere allows one to investigate the molecular mechanism of the biological system. So this we have to understand this molecular mechanism, and this involves the use of new chemical tools and techniques. So these are the innovations, and you have to use some concepts to design these chemical tools that I will discuss a little bit for the manipulation of visualization of key biological processes. And this analysis would in turn aid in development of potent chemical molecules for pharmaceutical importance or other industrial importance. So this part is mostly covered by Professor Majumdar, the gene identification, polymeric chain reaction, cloning, and then you are producing your sample for which you want to study. So this is mostly the protein. So methods in structural biology, if we look back, then we require the three-dimensional structures of macromolecules at atomic level. You see, atomic level, those chemistry students, they know atomic radiuses of carbon is about 0.77 angstrom, and the van der Waals radius of carbon is 1.7 angstrom. And whenever two carbons are coming, so they should not cross their van der Waals radius. So 1.7 plus 1.7 is 3.4 angstrom. So no molecule should come within 3.4 angstrom. If these atoms comes within this, either they have to make the covalent bonding through this atomic radii, or they cannot come inside except hydrogen bonding where some ionic interactions are there. So experimental information is combined with stereochemical and biophysical information to build consistent atomic models. So you have to correlate your function in biological and chemical information. So whenever you go do the pathological test, always you will see at the bottom of your report, it is written, this should be clinically correlated. So whatever the test report you have to clinically correlate all your results. That is also important part. You may get some structure, you do not know whether that is a functional structure or not. So whatever the structure you are getting, then you have to correlate with this consistent chemical nature of this molecule. And these structures can provide vital information about the underlying principle of human disease and opening new ways to treat them. Like you see, this vaccine is being developed for this COVID-19, and many methodologies, many concepts have been used, and all these are from the structures. They have also used these concepts. Normally, the size of macromolecules in tens of nanometers. So if you look for this COVID virus, you will see this is about 30 nanometer. That means 300 angstrom. The whole size, spherical size is 300 angstrom. For proteins, in a single protein molecule level, you will find 30, 40, 50, 60, 80, 100 nanometer, 100 angstrom level, their diameter. But whenever these molecules make assembly, like ribosomes, it's a kind of virus kinds of assembly, because large number of molecules you have considered, and then you are going for structure determination. So that's why they are in the tens of nanometers level. And but by our resolvable objects, by our eye, we can really resolve up to 0.2 to 0.4 micrometer. And you see our molecules we are thinking about in angstrom level, in 20, 30, 40 angstrom level. So whatever the microscope you have probably will not give you the resolution at this atomic level, in angstrom level. So that's why there are three methods currently now coming. X-ray crystallography, very well-known method, where the first protein structure in 1963 of myoglobin was published. Nobel Prize was given. Nuclear magnetic resonance also is another method, but there are limitations because it cannot deal with the high molecular weight proteins. Maybe in monomeric state, it may be 15 kilodalton. In dimeric state, maybe it is 30 kilodalton. So there is a limitation of this molecular weight, but you can really measure the dynamics of the interactions. And cryo-electron microscopy is very coming up. Already 2 Nobel Prize have come up in 2019 by Venki Ramakrishnan, the structure of ribosomes, and in last years also you will see. And they actually Venki Ramakrishnan got in chemistry, Nobel Prize in chemistry, and last year also from University of Cambridge, three members, they got this Nobel Prize for the methodology development, how to increase the resolution by electron microscopy method. So these are important parts. And whenever you are using this X-ray or electron microscopy, means electron beams, obviously there is a chance of radiation damage. So whatever the molecule you are working with, this will cause the damage in the molecular structure. And how can you cope up, or how can you handle this situation? So bleaching and stability of the molecules also you have to look into. And that's why this methodology development last year, they got Nobel Prize for this electron microscopy. So mostly this is, you are using cryo. That means you are working at 100 degree Kelvin, where the heat generated by these electron beams or X-rays are mutated by this cold condition of this 100 degree Kelvin. Okay. So now in cryo-electron microscopy, if you see, you don't have to really go through the hurdles of producing crystals. You need only purified proteins in a very small amount. You may not require very high amount. And then you have to get the sample, grid, and then put in the electron microscope. You will get two-dimensional projections. Then this two-dimensional projection means you are getting the particles. So these particles are oriented in different directions. You are capturing images, millions of images of these particles. And from these, by the methods as used in the crystallography and other mathematical Fourier transformation method, superimposition method, reconstruction of images, you will get a three-dimensional electron density map, wherefrom you can really get the three-dimensional structure of the assembly. And mostly important for these big molecules, which are really working army in the physiological system. So these are the basically some micrograph of some structure. Mostly this is the FAS1 fatty acid synthase 1 protein complex. This is a multi-domain single enzyme protein. And you see these are the particles, and these particles, if you see, these are the orientation of the particles. So you have to collect millions, more than millions images, because these particles are oriented at different orientation. So you have to capture and capture. If you can get large number of data set, then probably you can go up to 3 angstrom resolution from this electron microscopy structure. Okay, so from these you can see very, very big molecules, multi-domain protein structures have been solved by this electron microscope in cryo method. So now nuclear magnetic resonance, obviously there is something, because any molecule will not give you the NMR signal. It has to be paramagnetic, and it has to be C13 level, because mostly we get C12. It has to be C13 level or N15 level, and sometime we have to add both C13 and N15. So depending on this, we can take proton NMR, we can take C13 NMR, we can take N15 NMR to get the signals, and also we can combine all these things to get the two-dimensional or three-dimensional NMR signals. So just I am giving some example. So this is the molecule, original signals obtained, and whenever anything is interacting with this molecule, the atoms are moved actually, and this is the sievement. This is the sit in the peaks you can see, and you can really tell which are the amino acids involved in this interactions. So these are the residue numbers you are getting, and this is the change in the NMR signal, and measuring which residue actually changing most. Then getting X-ray crystallography, the main problem in X-ray crystallography is you have to select the protein, important protein, then you have to express and purify this protein in soluble form, in active form. Then you have to do crystallization trial. Actually, this is not in your hand to crystallize this protein. So as long as the protein cannot be crystallized, you can tell this is not really science, this is art. This is an art. But when protein crystallizes, then probably your science starts. So nobody knows actually which condition you will get the crystals of any protein molecules. So this is a trial method. And then you have to do optimization of the crystallization condition, because you have to get a particular size of the protein crystals so that you can mount it and get the structures, and then you can collect the data of the single crystals. So whatever protein you want to solve the structure by crystallography, it is not your wish. You can only try. Maybe after 100 trials, then you may give up the project even, since no crystals you are getting. Okay, so these are the crystals. They are very strong, because for crystallization you require very purified proteins which really sits in a particular array, because if there is any change in the conformation, then there will be disruption in the pattern of the molecules inside the crystal. That's why now the purity level, whenever we are talking about this purity of the proteins, it's not the purity what you can see in the SDS page gel. It is a purity in the conformational level. So you maybe have single protein entity, but let's say there are 1000 protein molecules within the solution, same protein molecule, but in different conformation, because there are many flexible loops. If there are different conformations or the flexible loops, they may not be in identical conformation, and hence they cannot be crystallized. So this part you cannot really find out what is the percentage of molecules are in the different conformation. So you have to get the crystals, then the X-ray diffraction it will happen. So it is not really microscope. So here we are using fluorescent, this Fourier transformation. This is a mathematical lens we call. By Fourier transformation, we get the electron density like this, and then from this electron density, you can see all the carbon. This is the carbon. This is the carbon. This is the carbon. This is carbon. This is nitrogen. This is oxygen. So you see all these atoms are located, and when these atoms are located, then basically you are getting this kind of structure. So this is a three-dimensional fold of the protein. So you know it's not a straight line. In the primary structure of protein, it is just a sequence. In secondary structure of protein, it is just this helix or some beta strand. These are the beta strand. These are the beta strand. Or you will get some loops. This is loop. Okay, so loops are very flexible, and they fold in a particular fashion, and only then the function is achieved from these folded structures. So you have to go through this all purification processes. This is the SDS phase gel. You see, this is almost a single band, but still you can see some kind of impurities which may be difficult. And whenever you are running SDS page, basically you are denaturing protein and seeing whether there is only one kind of band or not. You see only one kind of band. It does not prove that it is not a mixture of the dimer, trimer or tetramer, because you have denatured the protein. So whenever this solution is found and done some side exclusion chromatography, then you may have concept of what is the probable molecular weight of this protein, whether this is in monomeric form, whether this is in dimeric form or trimeric form. Or you can also use some other methodologies like dynamic light scattering method. So it will also tell the particle size. But sometimes monomer, dimer or dimer, tetramer, they are in dynamic equilibrium. Even if you remove one monomer or one dimer, then automatically due to this equilibrium, they will also always convert it to another monomer or another dimer. So always you will get a mixture, and it's very difficult to handle this kind of protein. Whenever we are getting the structures of all these proteins, you seem that it is a monomeric structure or dimeric, but you do not really know what kind of dimeric assembly or monomeric assembly or tetrameric assembly, whether homo assembly or hetero assembly really interacting with other molecules or functional molecules. That is very difficult to understand, and for that sometimes we have to use the mass fingerprinting. Basically, this is called MALD, matrix assisted laser desorption ionization method, to find out what kind of molecules these pure proteins are interacting in the physiological system. So now come to some structures which I am interested in. Acetyl-CoA is very important molecules in the biological system. You can see the structure. Acetyl group is there, and CoA group, I will show the structure in the later part. So this is the coenzyme means CoA. So this is the CoA structure. You see the very long chain, and there is some sulfur. When the sulfur is attached to H, then it is CoA. When the sulfur is attached to COR, when acetyl group or acyl group, then it is called acyl-CoA. Then these molecules I will come in my later part, how this can be used, because this is a fluorescent molecule. This I will come with some kind of drug molecule, because this was used in 1989 or 1980s for tuberculosis treatment. But after that, it was seen that this has a very bad effect on the health. It causes teratogenic effect, and that's why this thiacetone was stopped for the TB treatment. And then thiacetone, basically a prodrug. It is not really a drug. Some enzymes works on this thiacetone, and then it makes the drugs, the sulfinic acid. For chemistry point of view, all these chemists, they should look into this conformation. So this C double bond is oxidized to sulfinic acid. And most of the cofactors, I have just given some example, NADH and NADPH. You see all the structures, and only you will find the difference in the phosphate group here. Okay, so this phosphate means you are adding some negative charges here. So these are the important molecules I will be talking about, and also this thing I will talk about. Just you see a chain having very long carbon chain, and these are mostly hydrophobic in nature. And also you will see some kind of modification here, and they are attached to some sugar molecule, and all these sugar molecules, these same kind of molecules are attached 2 times, and that's why it is called dimycolate. So this part is called the mycolic acid part. I will give you some more presentation on this mycolic in the latter part of my talk. So I will talk some proteins which are dehydratases, or some acyl carrier proteins, a protein that carries the acyl group, and some keto-reductases, and the drugs used, TB drugs used, thiolactomycin, isoniazid, and then ethanamide, pyrazinamide, and not used in thiacetone, isoxyl, and NAS. So I have given some introduction of all these molecules, and now I will go for the next slide. Okay, whenever we are talking about protein, so protein is, people know this is alpha amino acids. So this is alpha amino acid means there is only one carbon atom. So this is the carbon atom. This is called alpha with respect to the COOH group. So those chemists, they know how to do this nomenclature. And this glycine, since its carbon is attached with both hydrogen, the same kind of moieties, so they are not enantiomers. So you will not get any D or L of this glycine. But after that, all this R group, you will see the CH3, then valine, this isopropyl group, then cysteine, SH group. So you will see different kinds of R groups are attached, and hence we are only talking about the L amino acid. So in the biological system, the proteins are made up for higher eukaryotes in L amino acids, though you can find some D amino acids in the smaller organism, this prokaryotes organism. They may not be protein but something else, like gramicidin. If you go, then you will see there alternate L and D amino acids are there, and this is the ion channel basically, gramicidin, and that is also used as a drug molecule. So these are non-polars, because if you look at all this R group, then they are all hydrophobic in nature. But there are also polar groups where plus and minus charges also there. Some polar nature of the side chains are there. So I have to give some introduction of these amino acids, because these are the base, and these are the basic units of the proteins. And whenever these amino acids are connected with each other, they make the peptide bond. And mostly the CONH is a transpeptide, though because you have N lone pair and oxygen here, there will be resonance, and you will get mostly a flat time of peptide bond, where the hydrogen and oxygen may be mostly, in the cases they are trans in nature. But wherever there is proline, then they may be in the cis conformation. And if you see all this structure, any C alpha atom, if you see, so these are the single bond, and this is single bond, and single bond free rotation is possible, and they actually dictate the structure of the proteins. we are called, these are the Ramachandran angles, and they dictate whenever this Ramachandran angle are repeating in nature, they produce the secondary structures, and whenever they are random, they are only producing the flexible loops, but they cannot take any values, and that's why the Ramachandran plot was discovered by G. N. Ramachandran, the Indian biophysicist, which actually still today is very valid and the basically basis of the structure validity. Okay, so now I come to this tuberculosis. So if you look for this tuberculosis, they have the membrane, and they have plasma membranes, then periplasmic, then your arabinogalactan or this peptidoglycan, and then some capsule and outer membrane. So this outer membrane is very important. So this is the part outer membrane where mycolic acid and TDM, what I have already given some introduction in the last slide, where dimycolate is there. So these are the thick outer membrane layer through which any drug cannot pass. So if we can really stop the formation of this mycolic acid, then probably the Mycobacterium tuberculosis cannot grow or cannot really stable, and the drug permeability also will be very high. So there are hundreds of enzymes, 250 enzymes in Mycobacterium tuberculosis. There are 250 enzymes involved in this mycolic acid synthesis. I will show you only few of them. So whenever we are talking about what is mycolic acid now, so if you see this mycolic acid, this is the alpha carboxylic acid, beta hydroxy group. And with the alpha carboxylic group, there is about 26 carbon chains. There are 26 carbons are available. And in alpha mycolic acid, this is about 56 carbon chains are there. Okay, so there are 2 carbon chains attached, and they are very hydrophobic in nature. And that's why your outer membrane is so thick. And they are modified, they are methoxy or they are keto groups also there, and they make this Mycobacterium tuberculosis more virulent. So now I have given you some concept about what is mycolic acid and why mycolic acid is important for Mycobacterium tuberculosis and for drug designing. So this role of mycolic acid in regulation of biological processes in Mycobacterium tuberculosis, basically in cell synthesis, in survival of extreme condition, intermediate, impermeable to drugs, virulence, biofilm formation, so that they can really survive inside the macrophage, and all these things are rendered by mycolic acid. So this is a very important molecule in Mycobacterium tuberculosis. So mostly you will see there are 2 systems. Phase 1, I have shown you the Electron Microscopy structure. This phase 1 is a multi-domain single enzyme, and it starts with acetyl-CoA. In some point of time, this acetyl-CoA will be converted to malonyl-CoA. But through this phase 1, you will get mostly this C16 to C18 acyl-CoA. So this is normally the function of phase 1, which is mostly present in human system. But you will get phase 2 also in human mycobacteria. And this phase 2 is mostly present in Mycobacterium tuberculosis. Yes. So now there are other processes where this phase 1 will synthesis 24 to 26 carbon chain. So this is one process. So you see phase 1 can produce both C16 to C18 and C24 to C26. But still today we do not know what controls this ratio of C24 to C16. For normal human system, this C26 is produced in the body, and that's all. It's done. But in Mycobacterium, what happened? They hijack this phase 2, hijack C16, C18 molecules from phase 1 system. So phase 1 is basically in Mycobacterium tuberculosis producing both C16 and both C24. C24 is kept aside, and C16 will enter into phase 2 system, and this phase 2 system with some enzymes, this is MT5H, it enters into the phase 2 system, and this malonyl-CoA will be transformed into acyl carrier protein. You see, this is acyl carrier protein. So from CoA, Now this group is transfer, acylase is transfer here. So this is the function performed by this Mycobacterium tuberculosis FAB. This is another enzyme. And whenever this malonyl ACP, now you see in phase 1, this is CoA dependent, but in phase 2, it is ACP dependent. So this part is very important. This empty fat D is very important because this malonyl part is transported to this ACP part, and now this ACP malonyl ACP and C16 C18 acyl CoA, they will come interact with each other through Claisen condensation. Those chemists, they will understand what is Claisen condensation. They can compare this thing, and through this first enzyme, they produce this ketoacyl ACP. Then there is another enzyme MAP-A, which will see the CO group will be reduced to OH group. So all these reductases. Then OH group by HAD-AB, HAD-BC. There is a system in operation system that making a double bond. And after this double bond, this INHA, this double bond is reduced and making acyl ACP. So if you see actually past cycle, there is 2 carbon addition in the lipid formation. Okay, so every cycle it is giving 2 carbon extension in the lipid. So this is very important. So basically, if you start with C16, it ends with C18. If you start with C18, ends with 20, 20 will end with 22. In this way, it will go until it is 56 to 60. But in chemical, if you want to work this thing in your laboratory in a test tube, it is very difficult whenever this reaction are being done to in a controlled manner. Let's say you want just C30. Can you do it? Probably you cannot do it. So there must be some processes, some structural alignment so that which controls the entry of this long carbon chains. And this way you can produce really different kinds of lipids with different length specifically. And that's why you need the specificity and selectivity in the reactions. So these enzymes act as catalyst, biological catalyst, and they are very specific. Your product is very selective, and hence you can really use this thing for future work. So now for the point, phase 1 you have got C24 to C26, and phase 2 you have got C56 to C56 to C60. And for polyketide synthase, there is another enzyme whose structure is now determined by this electron microscopy. It's a big assembly, and by this way you can produce these mycolic acid. These 2 chains, one is 26, another is 56. These 2 chains are produced, and these are all synthesized in the cytoplasm, in the cytosol. And there are some transporter molecules which will transport into periplasmal membrane to cell wall and to outside through antigen 85C, which is a very good diagnostic marker for tuberculosis. So if you understand this basic part of this protein, of this mechanism, then probably it will be helpful to do the next part. So these are the drugs. If you see these are the drugs, these are the drugs, and these are the drugs, and these enzymes are the target molecules. So these drugs actually interact with all these enzyme molecules, and we will work mostly on INHA, HAD-AB, HAD-BC. We will focus on that. So using this structure, we will go for structure-based drug design. Probably time will not permit much. Then probe design, I will cover this part. And synthesis of the fatty acid derived chemicals, which will be the future focus for bioenergy. These different kinds of lipids are very important for bioenergy. If we can really specifically synthesize all these small molecule lipids specifically, then this will be used in bioenergy production. So mostly I will concentrate my talk today on probe design. So this is beta-catyl ACP. This is ACP part. By H2O release, then ion double bond is formed. So this is a kind of reduction. So there are many proteins are involved. I have named few proteins, and I will show in one operon, this HAD-ABC, how they are important for such kind of things. So here I will improvise some concepts, and let's see whether these chemists accept these concepts or not. So they are in same operon, means in the same operon there are 3 proteins, but they work only in dimer, either in had AB form or in had BC form. And you now understand, whenever this had AB or had BC, that is the rate limiting step actually. So had AB will decide or had BC will decide whether this mycolic acid formation will be involved in the persistence or in the virulence or in the pathogenicity. How these long carbons are incorporated inside the protein, that we will discuss little bit. So whenever we have got the structures, so we have got the structures of had AB in the complex form and had BC in the complex form. And now if you see in had AB and in had AB, this is the had one enzyme and had B is another enzyme, and they are making a complex dimer. And if we compare the structure, you see the structure are almost similar. You can really superimpose these two structures and hardly will find any differences. But if you look clearly, then you will see that active site in had B is aspartate 36 and histidine 41. So these 2 amino acid residues are involved in the catalytic process. Wherever in the same position, if you look for had A, then it is phenyl 39 and asparagine 34. So these are not really involved in the catalytic process. So why you require had A? That information was not so far known. So you solve the structures and we get the complete dimeric structures, and this had A is shown in green or cyan color and in had B in this black color. And we see that in had B has the catalytic residue, whereas had A is not. And there is two-fold screw axis between this had A and had B. And there is a group form where actually this thiacetazone, the band drug, can go inside. And what is the function of this thiacetazone? If you see, this is the active site group. So ideally, what should be entered here? This long fatty acid chain. So long fatty acid chain should enter, and these residues, these active site residues will work on the COH in the structure and make the double bond. That means they are interacting with this. So your long carbon chain should come through this path. So in some way, if we can stop the incorporation of this long fatty acid chain here, then further increase or further synthesis of this fatty acid or mycolic acid will be stopped. So then it is very much required that what are in the group. If you analyze this group, you will see they are mostly hydrophobic in nature. And you see these C26 or C30, C40 carbons, they are all hydrophobic. And whether they are a single chain hydrophobic chain or the chain is multi-folded, that also depends on the pathogenicity of the organism. So still so far, it is not really experimentally proved whether when this substrate enters into this group, whether they are in a single chain substrate, single straight chain is incorporated here, or the chain is multi-folded and incorporated here. Anyway, if it incorporated here, then the synthesis or catalytic mechanism will take place. But if we can stop the incorporation, then there will be no catalysis process. So this thiacetazone with this S, basically there is another enzyme I will come up. This will make this S into SOH. This will come here and you will see there is a C16. There are actually 2 cysteines. Here is C16, another cysteine is there. So this C61 will make disulfide bond with this thiacetazone sulfur and make a barrier kind of thing here. So once the barrier is made, then the chain cannot be incorporated here, and further synthesis of this long fatty acid is stopped. So mycolic acid synthesis stopped, and that's why this thiacetazone works. Now let me get into details little bit. So this is thiacetazone, and ETH, one is monooxygenase, because this is the precursor of the drug. This is we call prodrug. On prodrug, this monooxygenase of Mycobacterium tuberculosis works and make the sulfinic acid SOH here, and this SOH make the disulfide bond, one is C61 and C105. So this SOH makes C61 disulfide bond, and hence the entry point is this one, entry point is closed. No substrate can be incorporated here. Hence your mycolic acid synthesis is stopped. And as long as there is no thiacetazone, then both these sulfur C61 and C105 are open, and they make disulfide bond within this thing, and as a result they stabilize this structure. This had a structure, and this substrate can incorporate it from this process. So you understand now, after solving this structure, we understand the mechanism of HAD-AB. Similarly, HAD-BC, I am not talking about the HAD-BC now, so I am just concentrating on HAD-AB. Now, except from, because whenever you are solving the crystallographic structure, basically it is a snapshot structure. It is crystallized from in solid state form. So you want to know really exactly what is happening. So what is happening, if you want to see, then we want to see this ACP, means acyl carrier protein, on which this acyl carrier protein. So if you see this acyl carrier protein for the Mycobacterium tuberculosis, this is a little bit long. Long means if you see this part is extra in case of Mycobacterium tuberculosis. It has some role. This role we have to discover. And whenever you cut this 30 residue like E. coli, then E. coli is very stable structure. E. coli ACP is very stable structure. So E. coli ACP, if you do the molecular dynamics, now the role of the molecular dynamics, because we cannot really determine or see the structure movement in crystallography, we have to take some molecular dynamics tool, mathematical tools, so that we can really see the stability of the protein. So as long as ACP M82, means only 82 residues are involved, you see this is very stable. But whenever the whole ACPM, means C-terminal extra 30 residues are involved, you see lots of dynamics in the protein is found out. So why is this dynamics? We have discovered that there is a DSL motif and there is serine motif, and we have utilized this serine for some probe design. And you see this is for the whole protein, this green is for the whole protein. The dynamics very unstable, but for the small protein, this 33 residue truncated acyl carrier protein is very stable. It's very stable, whereas this part is very dynamics in nature. So come on. Now, so this is the acyl carrier protein, and I call this DSL motif. So this is the serine OH. What we have done actually, we said, this is the concept. So as per the title of this webinar, I have taken this concept and trying to give some light on it. So this is a serine OH. OH is active group. With this serine, you see this is exposed outside from the structure. You can see it. And with this, we have attached some pantothene group. So this is the pantothene group. And with this pantothene, whenever there is only pantothene, then we are calling this is a halo ACP. One is apo. Apo means there is no pantothene group is attached. Means absent of the ligand. And whenever ligand is attached, this is a hollow with ligand. So if we can attach, there is some purpose of this attaching. There is active side sulfhydryl group. Active sulfhydryl group. So we want to utilize this active sulfhydryl group here, and with the sulfhydryl group, we will attach some kind of fluorescence probe. So it will give you fluorescence. And what kind of fluorescence probe is this? This is a solvatochromic, means this fluorophore, if it interacts with the solvent, then you will not find any fluorescence because it will be quenched with water. If it is not with the solvent, if they are within the hydrophobic plate, then it is not exposed to the solvent, then you will get some kind of fluorescence. And then from that, you can really determine the microenvironment of particular protein. So you are trying to determine, so you have determined the structures of the protein, but I want to determine the dynamics and microenvironment of the protein attaching with this solvatochromic fluorophore. So this is 3-phosphoadenosine, this SH, and this is the molecule I already introduced before. This is called NBD iodoacetamide, and this is the chemical reaction full of chemistry. Then NBD coenzyme is formed. This is the CoA part, and with this CoA, this NBD is attached with this chemical reaction. And now we have attached this ACP, and now ACP, acyl carrier protein, is now attached with this pantothenic acid moiety and with this fluorophore probe. Now you see, whenever we are taking the mycobacterial acyl carrier protein with this NBD tag, we see a green color because it fluoresces, and this green color because it is definitely within some hydrophobic core. And whenever we denature this thing, then the structure is lost of this ACPN. As a result, this probe is exposed to solvent, and you see there is no color. So here there is no color. Okay, here there is no color because the protein is denatured, and now this probe is also exposed to water, and there is no fluorescence. So we have used this thing, and we have seen the full ACPM. Where is this probe is going? So this is the probe, and you see here there is some extra 33 residue. That's why this is the block. So this is becoming very selective in nature. But in case of when 33 residues are removed, then you will get more prominent green color because the whole probe is within the crevice, because there is no selectivity here, and hence you will get more green color. So we have compared now how these side chains, because side chains are mediated by ACP and this pantothenic acid with ACP. So now we can tell that how these be introduced within this structure. Okay. Okay, almost I am time up. So this is the model we have developed. So NBD pantothenic acid for ACP 82, when 33 residues are not there, you see this probe is within the very much of this hydrophobic cleft. But whenever this 30 residue is there, full protein, then this probe is not going totally inside, but showing little bit inside, and that's why you are getting this green color, right? So this is the theory for work. I am not going further because time is up. There are some slides on how these drugs are designed and how these molecules, these long chains are accommodated within this cleft. I am not going there. So now this is the time probably when I should acknowledge my, these are the molecules I have synthesis. So I am not going further. So basically utility of this is structure-based functional characterization to understand the pathophysiology or pathobiology of this infectious agent, structure-based drug design to facilitate identification of the new lead molecules of the pharmaceutical importance, enhancing the yield of fatty acid derivatives by bioengineering of microbes. This engineering, it is not in the not in the large scale, it is in a micro scale engineering, and design of enzymatic machinery to facilitate synthesis of methyl or ethyl esters, lactones. These are artificially for industrial applications, including plastics, lubricants, and biofuels. So this message I want to convey to our participant, that whenever you are working, you should have some philosophy against the work so that it can be utilized in further, and it can be helpful for the society. So all this work been done with funding from IIT Kharagpur, SCRB, DST, DBT, and from all my well sets. Then DBT provided my X-ray facility at the IIT Kharagpur. Professor Amit Bosak, the very well-known biochemist and chemist who synthesized most of the molecules for drug designing, which I have not shown it here. And 3 students, they are highly involved with this determination of these mycolic acid synthetic proteins, this Devojyoti, Rupam, and Bina, and Devranjan, joint student of Professor Bosak, who actually really designed and synthesized many more molecules, which shows really promising for inhibition of this mycobacterial growth, and all my present and past PhD students for their support. Thank you very much. And just maybe you are interested. So this is the COVID, and so far there are 159 249 structures are solved for this virus SARS-CoV-2, and 19 PDB structures for this week. In 1 week, there are 19 structures were released. So these are the whole structures. Basically, you will see the structures of the glycoprotein E2, which interacts with ACE2, and from that, actually knowing the structures of the E2, this HCV papillomavirus, HCV vaccine has been designed, Also, all these vaccine trials, they are targeting also E2 of this COVID, the SARS-CoV-2 virus. So these are the whole structures. So structures have lots of implication, and really you can see the structure. Now you can believe the science, and this seeing not through the general microscope, it is through the mathematical lens using Fourier transformation method and to determine the structures at the atomic level. Thank you very much.
Thank you, sir. There are some questions.
Sure.
I
Please.
am taking one of the questions. It has been asked by Orpita Roy. Her question is, in case of cryo-electron microscope, do the biomolecules need to be coated by gold or platinum?
Not really, not here. Whenever you are doing the particle analysis for cryo-electron microscopy, you don't need to coat for gold or platinum. No.
Okay. And some other questions, sir, which we have already answered during the course of your speech. And we once again thank you very much for your kind cooperation. And we hope we shall get such type of cooperation from you in future. Thank you very much, sir.
Thank you very much. You can share my email address also. If any other questions from the participants.
Okay, definitely. Okay, thank you, sir. Now we have come to the final part of the technical session. Now we shall move to our third and final lecture by Professor Dr. Sushanta Rai Chaudhuri, Chief Basic Science, Basic Research and Molecular Biology, Sharaj Gupta Cancer Center and Research Institute, Kolkata. Before I request Professor Chaudhuri to present his speech, I shall take the privilege to give a brief introduction of Professor Rai Chaudhuri. Sir, are you telling something?
No, I'm just want to know whether my slides are showing or not. My screen is showing or not.
Your screen is showing.
Showing, right?
Okay.
You can see the whole screen. Can you see my whole screen?
Yes, sir.
Full screen? Okay.
Yeah. Okay, sir, let me give the introduction for the audience, please. Okay.
Please go ahead.
Now I am going to give a brief introduction of Professor Rai Chaudhuri. Professor Rai Chaudhuri is a J.C. Bose National Fellow, Chief Basic Research, Sharaj Gupta Cancer Center and Research Institute, former Chief Scientist, CSI and Indian Institute of Chemical Biology, former Professor, Academy of Scientific and Innovative Research, Mahatma Gandhi Road, Thakurpukur. Dr. Sushanta Rai Chaudhuri is a cancer biologist. He obtained his PhD in biochemistry in 1985 from the University of Calcutta, India. From 1985 to 1990, he did postdoctoral research at University of Pennsylvania, USA. Dr. Rai Chaudhuri was former scientist during 1991 to 2015 at the CSIR, Indian Institute of Chemical Biology, Kolkata, India, and currently Chief Basic Research, Sharaj Gupta Cancer Center and Research Institute, Kolkata. Dr. Rai Chaudhuri is among the first few who initiated genomic research in India. As a cancer biologist, he contributed significantly on the molecular understanding of the genomic instabilities and functional consequences of gain of function p53 mutations in human cancer. He has also contributed significantly in the area of cancer stem cell using oral cancer model. He has developed a microarray-based gene expression signature that distinguishes the aggressive subset of early stage oral cancer from non-aggressive one. Currently, he is developing algorithm based on molecular classifications of ovarian, endometrial, and rectal cancers for prognostic and therapeutic decision. He published more than 150 research papers and other articles. Dr. Rai Chaudhuri has 7 genomic data submissions, 1 patent, and 1 copyright to his credit. He supervised 24 PhD students. Dr. Rai Chaudhuri is the fellow of all 3 science academies of India. He was elected as President of Indian Association for Cancer Research during the period 2016 to 2019. He was awarded the J.C. Bose National Fellowship by the Department of Science and Technology, Government of India in 2007. With this short introduction, I shall humbly request Dr. Rai Chaudhary to deliver his speech. Please, sir.
Thank you very much. I hope all of you can hear me properly.
Okay, yes, sir.
Yeah. At the outset, I thank the organizer, the faculties, the governing body, members, principal of Kharagpur College for giving me this opportunity to reach out to a large number of students, which I understand spans from school level students to postgraduate level students. I also thank NASI for giving me this opportunity, and also very proud to be associated with NASI in this kind of social educational activities. I must add one more disclaimer that I am the chairman of board of directors of a startup company, ATGC Diagnostic Private Limited. The subject of this particular webinar series conference is chemistry meets biology. Professor Majumdar has eloquently described the 200 years history of the development of bioscience, life science, where we have now reached, the rich theoretical knowledge base on which we try to understand our all biological systems. Professor Das has very interestingly described the intricacies of biomolecular reactions and how one can determine the precise chemical interactions in biological system at the level of atoms. What I'm going to do is to tell you in a very popular way how all this knowledge are being now used in the clinic for the patient's benefit, for patient's treatment. As you can see, the title of my talk is Precision Medicine and Cancer Management. Obviously, you can understand that I'll be talking about some advancements in the cancer management, but what I'm going to try to introduce to you a very new concept which we call the precision medicine. I will more explicit on this as I move forward. As you know, the chemistry meets biology to understand the various biological processes. And one of those outcome of these biological processes is the disease. All human beings suffer from various diseases. As Dr. Majumdar said, there are different organisms. We are talking about human. All humans suffer from various diseases. If we look very closely to all these diseases, we'll see there are two driving forces that actually determines whether or not a given individual will precipitate with the disease. One is our genes, and the other is our environment. The intimate interaction between these genes and environment actually determines that what would be the disease processes. As you see, there are several diseases where gene has the main role, like the inherited diseases as discussed earlier, like hemophilia or thalassemia. Whereas there are some diseases where environment plays a very strong role. Of course, the motor vehicle accident is a totally environmental consequences. But even if you look at the infectious diseases, as we understand that the disease is caused by an infection, as we are now totally shaken by a viral infection in our hand. But also you see these infectious diseases are not entirely dependent on the infectious agent. As you can see, now you can actually read in the newspaper every day, even the COVID-19 disease, which is caused by the infection of a virus called SARS-CoV-2, actually not giving similar type of outcome manifestation in all individuals. So this is what really actually the very important pertinent question in biology, that why the different individuals for a given disease, the outcome is different. So there is a whole lot of diversity, heterogeneity in the disease itself. So that's why this subject is so much multidisciplinary. It requires the knowledge of physics, the chemistry, the biology, the mathematics, the statistics, and now I add the computer science, the computer languages. As we can see that artificial intelligence is coming in a big way to contribute in this process and to understand the various disease processes. Dr. Majumdar has very eloquently described the history of the development of various seminal discoveries or knowledge that we learned over the 2 centuries. And all these really culminate to a great discovery, great knowledge base that was described, that was ushered in the year of 2001, when the complete nucleotide sequence of the human genome was enumerated. As it was said, that we have 23 pairs of chromosomes. All chromosomes are made of DNA, and all these DNA together consist of 3.2 billion bases. And each of these nucleotide, their organization, their sequence in our genome, we now know. As I said, the disease is caused by the genes and environment. We need to know which genes are causing which disease. Because of this seminal discovery in human genome sequence, it is now very easy and simple to identify the genes that are responsible for a given disease. But what we really are still lacking is to understand how this gene is interacting with the environment for a given disease manifestation. So the other important terminology in my title is the precision medicine. Well, as you know, in the past, any individual who has suffered from a disease goes to the doctor. Doctor looks after the various symptoms, and those are the data for the doctor to use his intuition to put into action and diagnose the disease. However, now what we really do, the doctors try to find out some pattern, some diagnostic tests which tells that which kind of disease is this particular patient is suffering from, and we call it as a evidence-based medicine. This is what we are going on, phases that we are going on through. But what is coming in the future is that, as I said, no two individuals keep similar types of symptoms, similar types of interactions with the environment to get to precipitate the disease. So we need to study millions such information and use our various algorithms to distill the evidence very precisely, that how in a given individual, what is the cause, what is the necessity, how this disease is being progressed, and based on that the doctor will give the medicine which we call the precision medicine. That means that particular, based on that particular specific individuals, all informations that a medicine will be delivered. I will elaborate this thing again. So let us come to our topics concern the cancer. As you know, cancer is a very dreadful disease, of course, and we are always very afraid of the cancer. Now we are very bothered with the disease of COVID-19. But as you can see, the COVID-19 kills only 3% of its affected individual, but cancer kills many high percentage of the population regularly. So it's a very difficult disease. And what is most important with respect to cancer is that it's not a single disease. There are almost 200 different types of cancer are known. And also it is becoming clear that two types of cancers, of course, are very different, but even within the same type of cancer, two different individuals with same types of tumor may have different response to the various management processes. So what we try to do at present, use various molecular techniques to profile these patients using histopathology, immunohistochemistry, fluorescence in situ hybridization, polymerase chain reaction, DNA sequencing, many other technology that we have developed, we have learned over the years. We try to use those technology to stratify these patients based on various markers. We call it a various kind of diagnostic markers. It is a prognostic, it could be a prognostic markers. It could be markers that is predictive of drug sensitivity or drug resistance, or it could be a marker for the adverse event. So based on these various markers or various evidences, we now stratify our patients in different categories, and based on those stratifications, different kinds of medicines are being prescribed for different patients. We call it a personalized cancer therapy. Now, precision oncology demands more. What it really demands is that it is not that just you match the patients with the therapy. It is also very equally important that you avoid the therapies that will not work. It's very important, as you can understand, cancer therapy is a very difficult therapy, very toxic therapy, and very costly therapy. So you really have to find out, you have to really have the markers in your hand. And I will give one example for such that who should give you what kind of therapy, because not all therapy will work equally to all cancer patients. Now, in order to understand that intricacies, that complicacy, we really have to do lots of basic biological research. Biology is very complex, as you can see. Now everything, all other curriculum are preceded by bio, because every people are now coming into biology to understand its complexity. The genomics, the imaging, the clinical laboratory techniques are few such knowledge base or technology base that are now being used in determining the precise nature of a disease in a given person. Now I give first a few examples that how this knowledge that we have gathered over the last 2 centuries are being used daily, regularly in the clinic for the benefit of the cancer patients. One of the great discovery for the cancer management is the development of a drug called imatinib mesylate. This particular drug is used for patients of leukemia, a specific type of leukemia which is called CML, chronic myelogenic leukemia patients. What happens in these patients is that because of a chromosomal aberration that I will discuss in the tell you in the next slide, a new protein which is not present in the normal blood cell called BCR-ABL, a fusion protein, is generated in these leukemic cells. And this protein is responsible for the high proliferation of these leukemic cells. So what people have done, this BCR-ABL is a kinase protein enzyme, so it requires an ATP molecule for its function. So what people have done, designed a molecule called imatinib mesylate, which specifically binds to the protein where the ATP molecule binds, thus not allowing the ATP molecule to bind. As a result, the BCR-ABL oncogenic protein, the fusion protein, cannot function, and this molecule has made a great development, great advancement in the treatment of this type of CML patients. Using this drug, these patients can now live decades, if not completely cured. Now, what kind of chromosome abnormalities made this thing happen? Actually, there is a specific translocation between two chromosomes, chromosome 9 and 22, between the two genes ABL1 and BCR. Because of these translocations, a new chromosome is generated where these BCR and ABL genes get fused together, and a new chromosome is generated, which we call the Philadelphia chromosomes. Now, upon these translocations from this fused gene, there are more than one transcript, the RNA molecules are generated. These two transcripts are generated in 95% of chronic myeloid patients. These transcripts are generated in 5% and 15 of the pediatric and 15 to 30% in the adult leukemic patients, as you can see here. So, when a leukemic patient comes to a clinician, he or she has to decide, looking at his blood profile, whether this patient is suffering from what type of leukemia. And with some prior knowledge, they can predict, might be this patient has is suffering from a chronic myelogenous leukemia or acute lymphocytic leukemia. Now, when clinicians suspect that, clinician asked for a test, a very simple PCR-based test, whether this patient leukemic cells has this transcript or this transcript. And immediately that simple PCR-based analysis diagnose the patient either with this either of these two transcript or this transcript, and accordingly patient is confirmedly diagnosed that he or she is suffering from CML or he or she is suffering from ALL. And accordingly, again, when it is found, the clinician will prescribe the imatinib mesylate for its treatment. So patient receives this drug and lives happily for several years. But unfortunately, it has been found in a small percentage of these patients who are receiving this drug, they develop resistance against those drugs. And it was found that the resistance mechanisms of resistance against this drug is mostly a secondary mutation, another mutation in the same BCR-ABL gene. Or that is the major case, but there are also cases where some other reasons are also there. But now I will show you 3 cases that we have analyzed in our laboratory at Swaraj Gupta Cancer Center and Research Institute, where our molecular analysis is helping the patients and the doctors to make these patients live longer. Here is a patient who was diagnosed with CML in 2004, and she was given imatinib and she was happily lived several years until in 2018, clinicians noticed that she is developing resistance against the imatinib. So she was referred to our laboratory, and we analyzed again that BCR-ABL gene by two different techniques. One is called the Sanger sequencing, and another is called the next generation sequencing. Professor Majumdar has told you that Frederick Sanger first discovered, described, developed the sequencing of DNA, and that method is called Sanger technique. And now subsequently, as I said, the whole human genome was sequenced by the same sequencing technology, but there was a great advancement in the technology that was developed by Sanger, and there is a technology that was used for the human genome sequencing. Sanger sequencing is low resolution sequencing. The NGS sequencing is a high resolution sequencing. Even a few genes, DNA molecules are present in a huge background of unmutated, non-mutated DNA also can be detected in the NGS technology, through NGS technology, which is not possible through Sanger technology. So we used both these technologies, the Sanger and NGS, and found that this lady has developed a secondary mutation at the 359 position. And as soon as we identified this mutation, it was a great relief for all of us, because there is another drug molecule that is available which can be now against this particular mutant BCR-ABL and can be given this patient, and who will again survive disease-free condition for several years. Now the second patients where again another guy in 2005 was developed the CML. He was given the imatinib. He was happy, and he was again developed resistance. So again the sample came to our laboratory. We analyzed its DNA, but now this time the Sanger could not detect any known mutations. However, the NGS, which is high resolution sequencing, can detected a less frequent mutation G250E. And as soon as these mutations was detected, again we are very relieved, because now this person can be given another drug which can work against these mutations. Now I come to my third case, where lady developed a mutation disease in 2010. He was given imatinib. She was happy. She was then developed again resistance, and again samples came to our lab, and we analyzed the DNA into this platform. But now in this case, we could not detect any known mutations against which the drugs have been developed. But instead, there is another novel mutations that have been detected, and it appears that mutations also have some functional consequences. Now, it is a matter of research to again understand how these mutations is involved, making this cell resistance to the imatinib, and how the new drugs can be developed against those mutations. So it came into the research domain. So as you can see, using very simple PCR-based techniques, we can detect, and what kind of disease it is, leukemia it is, and then we can give the drug accordingly. And if a person develops resistance, again, what can be whether we can give another drug to the resistance individual. Now I turn to another very important, frequently used therapy in another very dreadful cancer, which is the lung cancer. It is known that in lung cancer, many patients suffers mutation in a very important growth factor gene called epidermal growth factor receptor. This receptor molecule has three domains. One is the outer domain where the EGF molecule binds. One is the transmembrane domain, and one is the cytoplasmic domain. This cytoplasmic domain has a tyrosine kinase activity. When EGF molecule binds, it dimerizes, and this tyrosine kinase activity autophosphorates this domain, and the EGF molecule become active, receptor become active, and it sends signals inside the cell. Now, what happened? This will happen only when an EGF molecule will bind to the receptor. However, it has been found when they suffer a mutation in this particular domain, this tyrosine kinase is constitutively active. It really doesn't require EGF to bind. So as a result, it gives continuously the proliferation signal inside the cell. Also, there is another group of patients who have been found. The number of EGF receptor molecule has increased on the cancer cell surface. As a result, the continuously cell signal is going on. Now, looking at these two scenarios, in case of mutations where the tyrosine kinase domains got mutated, there is a drug molecule were developed, erlotinib or gefitinib, similar to as the imatinib that I discussed, where this molecule will bind to the ATP binding pocket of this tyrosine kinase domain and will inhibit the tyrosine kinase activity. As a result, signal will not proliferation signal will not go, so the cancer cell will not proliferate. It will stop proliferating and it will die. Similarly, there is antibody drug available called cetuximab, which can bind to this receptor and doesn't allow it to be activated by the EGF molecule. So both these types of drugs are now available in the market and regularly given to the patient. So the great knowledge of the discovery of the epidermal growth factor receptor as an oncogene and then development of all these drug molecules has ensured a very new, very effective treatment in a particular type of lung cancer. Now again here, in the patients of the lung cancer, this particular type of lung adenocarcinoma, there are 29 different mutations are known to occur in 4 different exons of this gene, and they could be a missense mutations or they could be in a deletion or insertions. Again, using a very simple PCR-based technique, we determine for a given patient which mutations are there, and based on those mutations, the clinicians gives various drugs to this patient, and they survive a longer time. But again, as in the previous case, again after giving this drug, these tumors develop resistance, and as has been found, one of the resistance mechanism is because of the secondary mutation T790M. The people have developed a drug against this T790M. So if somebody develops a resistance against this due to these mutations, then the person will give this drug. So as you can see, there is a constant war is going on between the cancer cell and the scientist. We are trying to unravel various various ways to overcome the cancer cells that the problem that gives us before us. So this war is continuously going on using the knowledge of chemistry, the knowledge of molecular biology, the knowledge of knowledge of cell biology. Actually, what have been found that these lung cancer patient, non-small cell lung cancer patient, actually of 2 types of adenocarcinoma and small cell carcinoma. The adenocarcinoma patients, when somebody is identified with the adenocarcinoma, and then the clinicians has 2 options. If they have a EGFR mutations, or they could have another mutations in another gene called ALK. if they fall into any either of this category, if the patient is suffering from EGFR mutation, as I said, they will be given the drug against anti-EGFR. But there is a caution. If the same patient has mutation in another gene called KRAS, then it is very unfortunate that this patient will not respond to this EGFR therapy. So for a patient, lung adenocarcinoma patients, to be effective with EGFR mutations, to be effective against the anti-EGFR therapy, should be EGFR KRAS positive. If he or she is KRAS negative, their mutations, then the therapy will not work. Similarly, if the patient has the ALK mutation, then instead of EGFR mutation, then they will be given another line of therapy. There is another drug called crizotinib, which is effective against this tumor. But if he doesn't have the ALK mutation, then it will not work. But if he has another mutation in another gene called ROS1, then again this drug will work. So as we can see, by monitoring 4 different genes, we can classify the lung adenocarcinoma patients to various categories and determine for which the anti-EGFR therapy will work and anti-EGFR therapy will not work, and for which ALK therapy will work, or maybe the ALK therapy will work despite of not having the ALK mutation but having the ROS1 mutation. So this is the algorithm that I was talking about, that we can develop by analyzing the genetic architecture of the tumor and deciding that which therapy will be best suited for a given patient. Now I turn to another important therapy that has come up recently. Is called using a very important principle called synthetic lethality. What happens, our cellular DNA is always get damaged, and we suffer lots of single strand break. But we are saved because there is an enzyme called PARP which repairs this single strand break. But if somehow PARP is not working, then this will be converted to a double strand break. Now, this double strand break is a very lethal for the cell. So what happens? We have another backup pathway called homologous recombination pathway, which repairs this double strand break of the DNA, and we survive. But unfortunately, in some patients, like BRCA1 or 2 mutations, like homologous recombination pathway gene mutated patients, BRCA mutation, as you know, BRCA gene is mutated many breast and ovarian cancer patients. In these patients, since they are a deficient of the HR, this will not be repaired. Okay, the cell will die. Now, using this particular strategy, what people have devised, that breast cancer patient who has the BRCA1 mutations, if their PARP is inhibited, then this single strand DNA will convert it to a double strand, and these tumor cells will die. And so there are lots of PARP, many PARP inhibitors have been developed as a drug and now given to the patients of breast and ovarian cancer for their treatment using this synthetic lethality concept. There is another very famous discovery that have been made in last few years. We have been always asked this question: why a tumor cell is not recognized by our immune system? Now, through the discoveries that received the Nobel Prize a couple of years back, we came to know that there are some proteins, receptor molecules that are present on the surface of the tumor cell, which prevents the tumor-specific T cell, the receptor, to interact with the antigen presented by the MHC on the tumor cell. So now the antibody have been generated which can block this interaction, so that now receptor can bind to this antigen and kill these tumor cells. This is called the immune checkpoint blockade or immune therapy. So because of all this discovery, now we have come to know there are genes which are mutated in the various cancer cells, and we can design molecules against those mutated cancer genes, which we call the targeted therapy. So nowadays it is very common that we take the tumor samples and do the mutational screening analysis to see what kind of mutation, which genes are mutated in a given patients, and using this various sequencing strategies, the Sanger sequencing, the NGS sequencing, using the NGS strategy, the various the genome, few number of genes or large number of genes or whole genome, various kind of sequencing strategies are now being developed, which are now coming into clinic to determine what kind of mutations are present in a given patients, so what kind of drugs can be given. Always it was our dream, like many of the disease, that we determine the presence of the cancer cell in the blood. As you know, generally the tumor, malignant tumor, is identified and confirmed through the biopsy of the tissue material of the tumor tissues. Sometimes it is very difficult and sometimes it's not possible. And so people always wanted to develop some technology where by testing the blood samples, we can identify whether a patient, a given individual has a growing tumor inside or not. And the technology is now being developed where you have come to know that these tumor cells, they secrete or they die because of the death. Lot of tumor cell materials are released in the blood samples, and using various techniques, those tumor released molecules like a DNA or the RNA or the protein, we are trying to detect either at the early level or at the localized disease level or at the metastatic disease level or at the refractory disease level. We're trying to find out whether we can determine at what stage of disease it is. It's our dream. The technology is being developed. Already for one particular disease, a technology has been marketed and people are using this is called the liquid biopsy technology, and more such diagnostic assays are in the clinical trial. We are hoping that they will be in clinics very soon. Always we have been developing drug molecules to combat various cancers, and for that purpose, what we do, we take the tumor cells. There are many tumor cells are there. We develop, we synthesize various molecules and screen those molecules against those cells that are grown in two-dimensional culture. And then subsequently, which molecule which shows some activity against killing activity of those cells, we test in animals. And then if it found to be good in animal, then we try to do the clinical trial. But unfortunately, through this strategy, it has been observed that more than 90%, not even 90, more than 95, 98% molecules fail when they are applied in the human clinical trials. One of the reasons it is believed because of the fact that it is when we do the initial screening, we do it in a cell growing in the two-dimensional culture, which is not the true scenario, true environment of a tumor inside a body, which is a three-dimensional in nature. So now it is now possible to develop the three-dimensional tumor outside the human body. We call it an ex vivo culture, or we call it an organoid culture. So now it is possible using a primary tumor tissue from an individual and grow it outside the body in a three-dimensional structure. Now we are dreaming. One way we will be these organoids will be used for screening various molecules, and we are hoping that they will give a better result. when they will be subsequently tested in the animals and humans. And there is another very big dream is there, that for the treatment is that when I take the tissue from a patient, grow in the laboratory, as well as will sequence in the tumor DNA and determine what kind of mutations are there. Now, based on those mutation profile, we'll screen a large number of drug molecules against those mutant proteins in the three-dimensional organoid culture, and hoping those which drug will show the sensitivity against this tumor of a given patient, those drugs will be prescribed to that patient. So as you can see, our dream is to go that kind of personalized character of a tumor to screen a drug molecule to see which drug molecule will be more effective for a given patient. We hope that this organoid technology will be available in near future for more precise determination, diagnosis of and also for clinical decisions. So as you can see, now the future is coming. There will be 4 stakeholders: the patients, the clinicians is already there, the clinical laboratories will do all the testings, and the researchers will take the information from the patients, and all their electronic health record will be taken. Their all molecular analysis will be done, and based on the database that we are creating for the millions of individuals, we will use that algorithm. Will generate a curated knowledge base, which will be now given to the clinicians on his computer desk, so that he or she can decide which medicine that or which treatment he will give to these patients. That's the dream. That's the precision medicines that are coming in future. I just complete with one of such study how these algorithms are developed. In oral cancer, there is in the early stage of oral cancer, there is a very important clinical questions. In the early stage of oral cancers, when the tumor is very small, they can be cured very easily by surgically removing that tumor. But unfortunately, about 30% of these patients some of these tumor cells are moved into the nearby lymph nodes. So if we remove this only, in those cases, if we remove this tumor only, then the cells that move into nearby lymph node will again form the tumor. But this happens only in the 30% cases. 70% cases, it doesn't happen. But because this be a possibility, surgeons, what it does, it removes this tumor as well as the nearby all lymph nodes, which is very morbid condition. So we thought, is it possible by using some gene signature, we can predict whether a given tumor tumor cell has moved to the nearby lymph nodes or not. So what we do, we took the tumor samples from 303 patients and 140 controls, and did the whole genome transcriptome analysis on the microarray platform, from which we got 2,980 genes which are differentially expressing between the tumor and the control. And then we applied the machine learning algorithms. We developed a machine learning algorithms using which we identified a 34 gene signature, which can more than 80% accuracy can predict whether a given patient has a tumor nodal metastasis or not. So by looking at this signature, we can the surgeon can decide whether he or she will go for an aggressive surgery or a conservative surgery. So this 34 gene signature is a diagnostic marker based on some computer machine learning based knowledge, which can be used for this clinical decision. This has been patented, and we hope that in future this will find in the clinic. So I end with by saying that this precision medicine, which is develops for an individual specific genetic profile, it will help in prevention, diagnosis, and treatment of many diseases. And I must tell that it has a huge market potentiality. With that, I end my talk, and I thank my colleagues at the SGCCI. I am so thankful to all these clinicians who are giving us this clinical acumen, clinical knowledge to us, which we lack as a basic scientist, and trying to amalgamate these two together to generate new knowledge in the area of cancer management. With that, I thank you all. Thank you very much for your patience. Thank you.
Thank you very much, sir, for your highly illuminating and informative speech. And all the speeches have already been praised by the participants, and many of the participants have requested to upload the matters. For the participants, I'd like to say that all the matters are now open on YouTube. Whenever you will click the YouTube link, then you will be able to listen to the lectures. There are some questions, sir. I am asking two of them.
Yes.
One question is, actually, it has been asked by one of our retired teachers of our department. He is around 80 years of age. Still, he is quite enthusiastic to listen to the whole program.
Thank you very much.
Professor Amitabh Datta Majumdar. He asked, is food habit responsible for cancer?
Definitely. I mean, obviously, there are many food habits which are not very good, and smoking habit, drinking habit, all these are environmental factors which can, influence the tumor formations. Yes, cancer formations.
Okay. And a second question, it has been asked by Himatri Das. He says, is there any change in treatment of medicine due to corona environment in cancer treatment?
Well, yeah, that's a big question. How much the COVID-19 is influencing the cancer treatment is now a very big issue. First of all, let me tell you, very unfortunately, the cancer patients who are succumbing to the COVID-19 infection are in a very poor condition because they are COVID-19 infected. The regular cancer hospitals, they are not able to get treatment from the regular cancer hospitals. They are going to COVID hospitals. Now, in many cases, initially it was proposed that say for a cancer patient, some chemotherapy is going on. Okay, so the chemotherapy was held back. Say for a patient, some surgery is supposed to be done. That surgery was held back. Okay, because of this COVID and the uncertainty with the COVID-19 situation. But now, it is becoming very apparent that we cannot hold back. As you can understand, you can wait for many other diseases, but cancer you cannot wait. As you lose one day, your patient is going more towards the bad condition. So people are now under this condition trying to treat these cancer patients. Now the question is that since COVID-19, the SARS-CoV-2 virus, one of the main cause for the disease manifestation is the hyperimmune reactions, that we call it a cytokine storm, and the immune system seems to have totally go vary with these viral infections. Okay, so now in cancer patients, because of these various, because their treatment like chemotherapy, radiotherapy, or because of general physical conditions, they have a very weak immune competence. Okay, so people are thinking, people thought that the COVID-19 will make real havoc in this cancer patients. Well, the data is still controversial. We are not still clear very much that whether the COVID-19 cancer patients with COVID-19 infection do better or do worse still more data is now to be generated to clearly understand that how they are. There are reports in both ways. Early reports from China and Italy says there is a very huge severity in the disease, but now some reports are coming which are saying may not be that bad. So it's we have to wait for some more time to really get clear picture.
Okay. Thank you, sir, once again very much for your kind informative speech and for sparing your very busy time with us. We are immensely thankful to you for this. With this, our technical session is over. Now we shall move to the valedictory session, but I would now request the participants and others to please stay with us, because whenever we decided to hold this webinar, we also decided to hold an online essay competition, state level essay competition, and almost 258 students from different parts of our state and as well as from different parts of our country participated there, and the results will be declared during this valedictory session. Uh, in the valedictory session, first of all, I'd like to request Professor Prashanna Dule, Head Department of Chemistry and Coordinator of the State Level Essay Competition, to address the audience. Prashanna, please. Prashanna? Hello? Prashanna?
Hello, Indra.
Ha. You are
Okay.
audible. You are audible. We can hear you.
Respected Professor Anjan Chaki, President of Governing Body, Kharagpur College, and Patron-in-Chief of the Webinar Organizing Committee, Dr. Vidyut Samanto, Principal, Kharagpur College, Chairman of Webinar Organizing Committee, respected resource person, Professor Dr. Hemant Kumar Mukherjee, Professor Dr. Amit Kumar Das, Professor Dr. Sushant Roy Chaudhary, and Sri Sandeep Sen, DSC member, HS Council. Dear participants, my colleagues, ladies and gentlemen, I welcome you all to the valedictory session of the webinar. It is a matter of delight that the Department of Chemistry, Kharagpur College, has organized an online state-level essay competition in collaboration with National Academy of Science India, Kolkata chapter, and a large number of participants have participated there. Today, the result of that competition will be announced by respected Sri Sandeep Sen, DSC member of HS Council, within 3 minutes. I, being the coordinator of the essay competition organizing committee, cordially welcome all the student participants to the result announcement ceremony. The essay competition was organized with the motto of the creating science awareness among the students of both school and college level. I come to know from jury members that the performance of the participants was really very much encouraging. I appreciate this effort of the participating student very much. Today's youth is the future of the nation. Through your participation and expressing your idea and thought about the present environmental condition and probable remedies, you all have shown your concern about the well-being of our beloved earth. I believe that participation itself is a great achievement, especially when National Academy of Science, India, Kolkata chapter has provided the platform. For this essay competition, we invite jury members from different colleges. Without their help, we cannot truthfully complete this competition. So I wish to thank all the jury members of this state level essay competition. Now I shall not extend my speech anymore. We are all waiting for the announcement of the result of the essay competition. With this, I conclude my speech. Thank you very much. Is your audio?
Okay. Okay. Thank you, Professor Prashanna, Head Department of Chemistry, for his address. Now I'd like to request Sri Sandeep Sen, Honorable DSC Member, HS Council, and Honorable Member of NASI and INSA, to deliver his speech. He has been the person who made it possible for us to make a very good coordination between Kharagpur College and NASI Kolkata chapter for organizing this state level webinar and the national level webinar and state level essay competition. For this, we are highly grateful to him. I'd request sir to kindly deliver this speech and announce the results as well. Please, sir.
Thanks. I simply would say about the webinar because the larger than all the scientists told everything clearly to us how biology and other subjects are related to this. In all good wishes to my friends, learn and getting this. The essay competition is successful and the juries are very tough to this task, but it's literally tied to all of the competitors. They are all very good, but there must be some results. I am going to declare the results. In school level essay competition, from upper primary to secondary and higher secondary, in 3 levels, students participated in this competition. And I am saying this is very much joyful. Pleasure to us. But first place goes to students in class 6. As I announced with them at the last, now I am announcing the school level 5th position is 2 candidates there. Shobha Ali from Al Amin Mission, Khalkapur, Howrah, class 11. Again I am saying Shobha Ali of class 11, Al Amin Mission, Khalkapur, Howrah. Another 5th position going to শোনা যাচ্ছে না? হ্যালো 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 হ্যালো
আপনার পিছনে ফ্যান বন্ধ করে দেন।
আচ্ছা
আপনার পিছনে ফ্যানটা বন্ধ করুন, তাহলে মনে হচ্ছে সাউন্ডটা ক্লিয়ার হয়ে যাবে।
Yes, yes.
It is a pleasure to us that first ঠিক আছে, it's okay, it's okay.
It's okay now.
Yes. It is pleasure of us that first position in school level essay competition is going on upper primary level students, and as I announced his name or her name at last, but at first I am announcing school level 5th position going to Soha Ali of Al Amin Mission, Khaltapur, Howrah of class 11. Second 5th position is to Anton Mistro of class 12, Kharagpur Alumni Polytechnic High School. Anton Mistro of 12, Kharagpur Alumni Polytechnic High School.
Atulmoni Polytechnic. That there must be some printing mistake. Atulmoni.
Yes.
Yes, sir.
Atulmoni Polytechnic High School. 4th position, Samuel Bhuya of Nimpura Arjo Vidyapit. Bhuya of Nimpura Arjo Vidyapit, Kharagpur, plus 12. 3rd position, Shatovisha Mitra of class 7, Gandhi Vidyapit, Egra. It is also from the upper primary level. Third position, Sotovisha, Gyandeep Vidyapit, Egra. Second position, Ipsita Acharya of Saint Agnes Higher Secondary School, Kharagpur. Ipsita Acharya of class 11, Saint Agnes Higher Secondary, Kharagpur. And first position, Anusmita Nath of class 6, Saint Joseph's Convent, Chandannagar. Anusmita Nath of class 6, Saint Joseph Convent, Chandannagar. Now I am shifting to college university level, and there is only fifth position I am telling first. Sneha Mohapatra of 4th semester Zoology, Kharagpur College. Sneha Mohapatra, 4th semester Zoology of Kharagpur College. Fourth, Devjit Mal, UG, 4th semester, Garbeta College. Devjit Mal, undergraduate students, 4th semester, Garbeta College. Third position, Joyita Sarkar, undergraduate 2nd semester of Presidency University. Third, Joyita Sarkar, undergraduate 2nd semester, Presidency University. And second position, Sushoma Mali, undergraduate 2nd semester, St. Paul's Cathedral Mission. Second, Sushoma Mali, BA 2nd semester, St. Paul's Cathedral Mission. And first position to Nathasri S, first year MBBS, MVJ Medical Research Hospital. Nathasri S, first year MBBS, MVJ Medical Research Hospital. And these are all the results. Thankful all the participants, my beloved students. You are very good, and we are happy. All the juries and judges are happy to go through your writings. Thank you all. Thank you, the Kharagpur College and all the coordinator, conveners, and others. Thanks from the side of NASI. Thanks from myself. Thanks.
Thank you, Mr. Sen, for your address and for taking the trouble to announce the results. We are grateful to you. Now,
Hello. Thank you, sir. I request coordinator Mr. Prasanna Dule to announce the results once more because some connection problem. So please announce the results once more.
Prasanna, please unmute your microphone. Prasanna, please unmute your microphone. Okay.
Okay, listen. For the school level, first, Anusmriti Nath, St. Joseph Convent, Chandannagar, class 6. Second comes Ipsita Acharya, St. Agnes Higher Secondary, Kharagpur. Third, Sotovisha Mishra, Gyandeep Vidyapit, Egra, class 7. Fourth, Shamal Bhuiya, Nimpura Arya Vidyapit, Kharagpur, class 12. Fifth, Soha Ali, Al Amin Mission, Khalagpur, Howrah Division, class 11. And one more, Ankon Mishra, Kharagpur Atulmani Polytechnic High School, class 12. For the college and university level, first is Nathasri S, first year MBBS, MBJ Medical and Research Hospital. Second, Sushoma Mali, BA second year, St. Paul Cathedral Mission. Third, Joyita Sarkar, UG second semester, Presidency University. Fourth, Devjit Mal, UG fourth sem, Garbeta College. And fifth, Sneha Mahapatra, fourth sem, Zoology, Kharagpur College. Thank you.
Thank you. Thank you, Professor Dule. Thank you, Mr. Sen, for your valuable speech. Now I would like to request Dr. Indranil Chakraborty, Assistant Professor, Department of Chemistry, Organizing Secretary of the Webinar Organizing Committee, to deliver vote of thanks. Dr. Chakraborty, please.
Honorable Patron-in-Chief, Chairman of the Seminar Organizing Committee, distinguished resource persons, delegates, my dear students, ladies and gentlemen. It is my honor and privilege to have been asked to propose a vote of thanks on the occasion of valediction of the national level webinar entitled Concepts, Innovation and Methods When Chemistry Meets Biology, organized by the Department of Chemistry, Kharagpur College and National Academy of Sciences, India, Kolkata chapter. At the outset, on behalf of the organizing committee and entire fraternity of Kharagpur College here together, and on my personal behalf, I extend a very hearty vote of thanks to all valued invited speakers, namely Professor Dr. Hemanta Kumar Majumdar, NASI Senior Scientist Platinum Jubilee Fellow and President NASI Kolkata Chapter, Dr. Professor Amit Kumar Das, Professor Department of Biotechnology and Head School of Biosciences, Indian Institute of Technology, Kharagpur, Professor Dr. Sushant Rai Chaudhuri, Chief Basic Research, Saroj Gupta Cancer Center and Research Institute, Kolkata, for gracing the webinar with their important works and sharing with us their findings and opinions. All the resource persons in the webinar are very busy persons working within a very tight schedule, but they have spared their time and they have remained present among us today in the webinar. We are in true sense grateful to them. I take this opportunity to thank Professor Anjan Chaki, Honorable President, Governing Body of Kharagpur College and Patron-in-Chief of the Webinar Organizing Committee for his all-round support and enthusiastic appraisal for initiating such type of academic programs. Ladies and gentlemen, Dr. Bidyut Samanto, Principal, Kharagpur College and Chairman of the Webinar Organizing Committee, has been the driving force behind the whole process. His vision, interest and passion has made it possible to hold a number of webinars, and this is one of those. I express profound thanks to him. It is our proud privilege to have been connected to the famous National Academy of Sciences India Kolkata chapter as a collaborator to organize the national level webinar as well as the state level essay competition. In this regard, Mr. Sandeep Sen, DSC member HS Council and member of NASI and Indian Academy of Science, who has made a successful and smooth coordination between NASI Kolkata chapter and Kharagpur College. We are in true sense thankful to him. He has also been an honorable jury member of the said essay competition. I express my deepest sense of indebtedness to the respected jury members, namely Dr. Bimal Krishna Das, Associate Professor, Department of Mathematics, Sri Rabindranath Chongdar, Associate Professor, Department of Commerce, Dr. Gagan Chandra Mandal, Associate Professor, Department of Chemistry, Dr. Jyotirmay Pramanik, Associate Professor, Department of Physics, Dr. Farid Sheikh and Srimati Koheli Pramanik, Assistant Professor, Department of Chemistry, Sri Kollan Shur, Sri Sanjay Kumar Bera, Srimati Labani Giri, State Aided College Teacher, Department of Chemistry, Srimati Moumita Chakraborty, Srimati Shivani Chaudhuri and Sri Abhimanyu Modi, State Aided College Teacher, Department of Zoology, Sri Ashutosh Chaudhuri, State Aided College Teacher, Physiology, Sri Sandeep Tripathi, State Aided College Teacher, Department of Geography, Dr. Shubhankari Prasad Chakraborty, Assistant Professor, Department of Physiology, Ramananda College, Vishnupur, Dr. Kaushik Ghosh, Assistant Professor, Department of Chemistry, Ghatal Rabindra Shatavarshiki Mahavidyalaya, and Dr. Shukesh Patra, Assistant Professor, Department of Chemistry, Midnapur College, for their valuable contribution towards evaluating the participants of the state level essay competition. Students from different schools, colleges, universities, and research institutes have participated in the state level essay competition. Along with the participants, I take this opportunity to express my deep sense of gratefulness to their teachers and guardians as well. A large number of participants from different academies, research institutes, industries, and many more from different corners of the world have joined the webinar. Most delightfully, I'd like to mention here that these participants have registered from different countries of the world. I, on behalf of the organizing committee, extend a very hearty appreciation to all of them. Ladies and gentlemen, an event like this cannot happen overnight. Dr. Prasanna Dule, Head Department of Chemistry, and Dr. Shubhra Mishra, Associate Professor of the same department, started rolling the wheel a month ago. It required planning and a bird's eye for details. Both of them took responsibility on their shoulders and started working day and night in indefatigable way. They are fortunate enough to be backed by a team of very motivated and dedicated departmental colleagues who know their jobs very well and are result-oriented. They all work smilingly, shoulder to shoulder, to make this webinar a grand success. Thanks to the two student participants for their interest and enthusiasm. Sri Amitabh Manna and Sri Himadri Shaha, students of fourth semester chemistry honors of Kharagpur College, have rendered their support in overcoming some technical difficulties. I thank them very much from the core of my heart. Dr. Gagan Chandra Mandal, the senior most faculty member of the department, as well as the associate professor of the department, is acknowledged for his valuable suggestions and encouragement. Dr. Jyotirmay Pramanik, coordinator IQAC Kharagpur College, is also acknowledged for constantly sharing technical knowledge for conducting online seminars like this. My special thanks goes to the Secretary Teachers Council, Dr. Bikash Kumar Ghosh. I'd like to thank Professor Tapan Kumar Pal, Honorable Member, Governing Body of Kharagpur College, for being with us from the very beginning. Accolades are also due to the members of Governing Body Kharagpur College and all my colleagues of this institution for their constant support. Last but not the least, I wish to extend my thanks to Sri Shrikanta Vashak, proprietor of Creative Mart Kolkata, for providing technical assistance for the last fortnight. I cannot thank everybody. I cannot thank everyone enough for their involvement and willingness to take on the completion of tasks beyond their comfort zones. It will remain evergreen in the memory. Thank you once again. With this, I officially announce closing of the seminar. Have a wonderful day ahead. The results have already been announced, and I'd like to convey official. I'd like to say to the participants that all the participants who have taken part in the state level essay competition will be sent the certificates. All the participants will get the certificate, participation certificate, but it will take time. We shall take 7 to 10 days for sending the participation certificates, and the awardees and the top 5 or 6 students whose names have been announced just now will get the special certificates from Kharagpur College as well as from NASI, and we shall communicate to you through your registered email ID. And the feedback form has already been posted probably in the chat box, and you were requested to send the feedback form and send it to the and submit it properly. And I once again thank you all the participants, delegates.
Please announce one thing, that physical prize also be present after the situation will become good. When the situation will from NASI.
Okay, I'm conveying one thing Mr. Sen has told, and we already told it, and we have already mentioned it in the brochure that NASI will also provide some physical prizes. They will provide some prize to the top 5 and 6 students of these both the school level and college level competitions. But during this situation, COVID-stricken situation, it's not possible to arrange such a prize distribution ceremony. The prize distribution ceremony will be organized after the situation is normalized, and then the position winners will definitely get some prize from NASI, and which will be communicated through your registered mail. Okay. Thank you all once again. Okay.
So I thank you very much.
Thank you, sir.
Yes, sir.
All organizers, a really great meeting. I enjoyed. Thank you for organizing this event.
It was our pleasure to have you with us, sir. And we have overcome all the problems, all the technical difficulties that we faced day before yesterday.
Yes, sir.
Yes, sir.
That I can.
But you have taken lots of trouble for us, sir.
Yes,
We
sir.
are really grateful.
So thank you very much.
Thank you.
Thank you, sir. Thank you, sir.
Thank you.
Thank you.